Prologue: The Cell That Opened the Wrong Door. Imagine for just a moment that you could shrink until an entire human body became a landscape, not a landscape of organs, not of bones, not even of blood. Instead, you would discover a civilization of nearly 37 trillion living cells. Each one is astonishingly busy. Each one is performing thousands of chemical reactions every second. Each one is keeping you alive without ever asking for recognition. Tonight, however, we are not beginning with a virus. That would be far too obvious. Every mystery has a victim before it has a suspect. So our story begins somewhere much quieter, inside a single healthy cell. At first glance, nothing seems unusual. Its microscopic factories hum with perfect rhythm. Tiny molecular machines assemble proteins one amino acid at a time, following instructions copied from DNA with remarkable precision. Every completed protein is folded, inspected, and sent to wherever it is needed, like carefully crafted tools leaving an impossibly efficient workshop. Elsewhere, thousands of mitochondria tirelessly generate ATP, the universal currency of cellular energy. Without them, muscles could not move, neurons could not think, and hearts could not beat. Every breath you take is quietly converted into the energy that powers this invisible city. Deep inside the nucleus, another crew is hard at work. The cell's DNA, more than 2 meters of genetic information folded into a microscopic space, is constantly being inspected. Every day, countless chemical injuries appear within this precious blueprint. Some are caused by sunlight, others by natural metabolism, still others simply by the passage of time. Yet highly specialized repair systems patrol the chromosomes without pause. They detect mistakes, cut away damaged pieces, replace missing letters, reconnect broken strands. If these molecular mechanics stopped working for only a short while, life itself would begin to unravel. Meanwhile, messages flow continuously throughout the body. Hormones arrive carrying instructions from distant organs. Neighboring cells exchange chemical signals. Immune cells briefly stop by, exchanging information before continuing their endless patrol. Nothing is isolated. Every cell belongs to a network so vast that no city humanity has ever built can truly compare. It is peaceful, orderly, predictable, a perfectly ordinary day. Then, something appears. Not a bacterium, not a parasite, not even something that most people would call alive. It is extraordinarily small. If a typical bacterium were the size of a school bus, this newcomer might be no larger than a basketball. It drifts silently through invisible currents, carried by a single breath, a tiny droplet, or perhaps a microscopic splash of fluid. No alarms sound, no explosions erupt, no dramatic invasion begins. The healthy cell continues its work, completely unaware that history is about to change. The tiny particle approaches the cell membrane. It does not roar. It does not attack. It does not announce itself. Instead, it simply waits. On its outer surface are proteins shaped with extraordinary precision. Not random, not accidental. Every curve, every fold, every molecular angle has been refined through millions of years of evolution. It is carrying something that resembles a key. And somewhere on the surface of the cell, there is a lock. Neither side understands the meeting that is about to occur. The cell believes it is simply interacting with another harmless molecule. The particle has only one purpose: find the right lock. Touch it. Open the door. The contact lasts only moments. No pain, no warning, no sound. If you could watch this event through the world's most powerful microscope, you would probably miss it entirely. Nothing dramatic happens. The cell continues making proteins. The mitochondria continue producing energy. DNA repair enzymes continue their careful work. Every process appears perfectly normal. But appearances can be astonishingly deceptive, because somewhere inside that seemingly insignificant particle lies a strand of genetic instructions. Instructions that did not belong to the cell. Instructions that the cell has never chosen to read. Instructions that, once released, may begin rewriting the destiny of everything inside. Minutes pass. Then something extraordinary begins. The ribosomes, the cell's protein-building machines, start receiving unfamiliar messages. Its resources slowly change direction. Energy is quietly redirected. Raw materials are consumed for purposes never intended by the cell itself. The factory has not been destroyed. It has been repurposed. Every machine is still running. Every worker is still working, but none of them are working for their original owner anymore. Without firing a single shot, without breaking a single wall, without killing the cell, the entire factory has changed hands. And perhaps that is the most astonishing part of our story. The greatest biological hijacking in the history of life rarely begins with violence. It begins with trust. A lock, a key, a door that should never have been opened. This raises a question that has puzzled scientists for more than a century. What exactly is a virus? Is it alive? It cannot eat. It cannot breathe. It cannot produce energy. It cannot reproduce by itself. Outside a living cell, many viruses are little more than carefully packaged genetic information, drifting through the world in complete inactivity. Yet once they enter the right cell, they awaken an astonishing sequence of events. They seize control of molecular machinery they did not build. They manufacture copies of themselves using resources they did not create. They spread across tissues, across bodies, and sometimes across entire civilizations. How can something that appears almost lifeless behave with such extraordinary precision? How can something incapable of living on its own command, one of the most sophisticated living systems ever to exist? Perhaps we have been asking the wrong question all along. Perhaps the real mystery is not whether viruses are alive. Perhaps the real mystery is how life itself can be persuaded to work for something that is not. That question has challenged generations of biologists. It has reshaped medicine. It has transformed our understanding of evolution. And it will guide every chapter of the journey we are about to begin. Because this is not simply the story of viruses. It is the story of invisible infiltrators, silent takeovers, molecular deception, and one of the greatest unsolved riddles at the very edge of biology itself. If you enjoy slow, immersive science stories that uncover the hidden mysteries inside your own body, please consider giving this video a like and subscribing to the channel. Our channel is still very new, and every subscription, every like, and every kind comment genuinely helps us continue creating thoughtful, carefully researched episodes like this one. Your support means far more than you might imagine, and we are sincerely grateful that you've chosen to spend your time with us today. Now, let us step even closer. Because before a virus can hijack a cell, we first need to answer a deceptively simple question. What exactly is a virus? Chapter 1. What exactly is a virus? Every great mystery begins with a mistaken identity. For centuries, humanity believed that every infectious disease must be caused by a living invader, something that hunted, fed, multiplied, and behaved much like any other organism we knew. It seemed obvious. When people became sick, something alive must have entered the body. But history has a habit of humbling certainty. Near the end of the 19th century, scientists investigating a strange disease that was destroying tobacco plants encountered something impossible. The infectious agent slipped effortlessly through filters designed to trap bacteria. Nothing should have passed through, yet whatever remained in the filtered liquid could still infect healthy plants. The culprit was invisible to the microscopes of the day. It could not be grown like bacteria. It behaved unlike anything anyone had ever studied. It was not until decades later, when electron microscopes finally allowed humanity to peer into a world measured in nanometers rather than micrometers, that the suspect finally revealed its face. And what a strange face it was. Not a cell, not an animal, not a fungus, not a bacterium, not even something that seemed fully alive. Tonight, we reopen that century-old case. Because before we can understand how viruses hijack living cells, we must first answer a question that still sparks debate even today. What exactly is a virus? Imagine entering a vast manufacturing city. Every building is connected. Every machine has a purpose. Electricity flows continuously. Workers communicate constantly. Supplies arrive. Waste is removed. Repairs happen without pause. That city is a living cell. Now imagine placing beside it a sealed flash drive. The flash drive contains instructions, very important instructions, potentially dangerous instructions. But by itself, it does absolutely nothing. It cannot read its own files. It cannot build a computer. It cannot generate electricity. It cannot copy itself. It simply waits. That comparison is surprisingly close to what a virus actually is. A virus is not a miniature cell. It is not a tiny bacterium. It is not a simplified animal. Instead, it is a remarkably compact package of genetic information. Inside nearly every virus lies one of two possibilities. either DNA or RNA, never both. DNA viruses store their instructions in the same molecule used by almost every living organism. RNA viruses choose a different language, one that is often faster but far more prone to mistakes. Those mistakes, as we shall eventually discover, may become one of evolution's greatest weapons. Surrounding this genetic material is a protective shell made of proteins. Scientists call it a capsid. Think of the capsid as an armored briefcase. Its purpose is not to think, not to move, not to breathe, only to protect the genetic instructions hidden inside. Some viruses possess an additional layer, a fragile outer membrane stolen from the cells they previously infected. This is known as the lipid envelope. Ironically, many viruses disguise themselves using pieces of their former victims. Like a master spy wearing a stolen uniform, they appear less suspicious as they approach their next target. But even with this disguise, something enormous is still missing. Everything that defines cellular life is absent. There is no nucleus directing operations, no ribosomes assembling proteins, no mitochondria producing energy, no ATP reserves waiting to power chemical reactions, no metabolism converting nutrients into fuel, no waste management, no internal repair systems, no independent communication, no ability to respond intelligently to changing environments. In fact, outside a living cell, a virus performs almost no biological activity at all. It does not consume food. It does not grow older in the familiar sense. It does not become tired. It simply exists, waiting. Sometimes for hours, sometimes for days, sometimes for years. Scientists have recovered viruses from Arctic ice that remained infectious after astonishing lengths of time. Others persist quietly on surfaces or within dormant hosts until the perfect opportunity appears. They are less like travelers on a journey, and more like encrypted messages waiting for the correct computer to decode them. Perhaps that is why the flash drive analogy is so powerful. Imagine receiving a USB drive containing malicious software. By itself, the device is harmless. Leave it inside a drawer for 1,000 years. Nothing happens. But connect it to the right computer, and suddenly everything changes. The computer itself begins executing instructions that never belonged to it. Files are copied, systems are altered, resources are redirected. The machine unknowingly becomes part of someone else's plan. A virus behaves in much the same way. Its genetic code is the software. The living cell is the computer. The cell provides every processor, every source of electricity, every construction worker, every manufacturing line. Without the computer, the malicious code is powerless. Without the living cell, the virus remains astonishingly inactive. This peculiar dependence has led scientists into one of biology's greatest philosophical debates. Can something that cannot reproduce on its own truly be called alive? Living organisms usually satisfy several expectations. They consume energy. They maintain internal balance. They grow. They respond to their surroundings. They reproduce independently. Viruses accomplish almost none of these tasks alone, yet once they enter a suitable cell, they suddenly participate in one of the most successful reproductive strategies on Earth. So where should we place them? Some researchers argue that viruses are not alive. Others suggest they occupy a unique biological category entirely separate from traditional life. Perhaps life is not a simple on or off switch. Perhaps it is a spectrum. And viruses exist precisely along its mysterious boundary. That boundary becomes even stranger when we examine the astonishing diversity of viruses themselves. Not all viruses look alike. Not all infect humans. Not all even infect animals. Some specialize in bacteria. These remarkable entities are known as bacteriophages, often shortened to simply phages. If any virus resembles something from science fiction, it is the bacteriophage. Its geometric head contains genetic material packed under extraordinary pressure. Below it extends a tail equipped with microscopic fibers that recognize bacterial surfaces with incredible precision. When the correct bacterium is found, the phage anchors itself like a lunar landing craft before injecting its genetic instructions directly through the bacterial wall. It is an engineering marvel built entirely from proteins. Then there are coronaviruses. Unlike the robotic appearance of bacteriophages, coronaviruses are wrapped inside delicate lipid envelopes decorated with distinctive spike proteins. Those spikes gave the coronavirus family its name because, under the electron microscope, they resemble the glowing crown, or corona surrounding the sun during an eclipse. Influenza viruses appear different again. Their surfaces are covered with two famous proteins, hemagglutinin and neuraminidase, tiny molecular tools that help the virus enter cells and later escape to infect new ones. Every seasonal flu epidemic depends upon these microscopic structures. Then comes HIV. Unlike many viruses, HIV carries an extraordinary enzyme called reverse transcriptase. Instead of following biology's usual rule, DNA makes RNA, HIV performs the process in reverse. Its RNA is converted into DNA, allowing the virus to integrate itself into the genetic library of the very immune cells sent to destroy it. The hunter becomes the hiding place. Herpes viruses have perfected another strategy. Rather than multiplying continuously, they disappear. Following an initial infection, they retreat into nerve cells and remain almost completely silent. Months later, years later, sometimes decades later, they emerge again, as though waking from an invisible sleep. The infection appears to have ended. The virus knows otherwise. And then there are adenoviruses. Unlike influenza or coronaviruses, adenoviruses possess no fragile lipid envelope. Their sturdy protein shells allow them to survive far longer in harsh environments, helping explain why they spread so effectively through contaminated surfaces and close human contact. Different appearances, different strategies, different hosts. Yet every one of them shares the same extraordinary limitation. None possesses the machinery necessary to live independently. Each must borrow life from something already alive. Perhaps that is the most unsettling discovery of all. Viruses are not conquerors because they are stronger. They are conquerors because they are astonishingly efficient thieves. They steal energy they never produced. They borrow factories they never built. They exploit workers they never hired. Every protein they manufacture is assembled by someone else's machinery. Every copy of themselves is constructed inside another organism's home. Seen from this perspective, a virus is less like an independent organism and more like a perfectly written set of biological instructions searching endlessly for a machine capable of reading them. It is information wrapped inside protein, nothing more, and somehow nothing less. Which brings us back to the mystery introduced at the beginning of our journey. If a virus cannot generate energy, cannot manufacture proteins, cannot reproduce by itself, cannot even be confidently described as alive, then who is truly making the millions of new viruses that appear during an infection? The answer may be one of the most astonishing revelations in all of biology, because the virus does not build them. The infected cell does. And in our next chapter, we will discover how an apparently healthy cell can mistake an intruder for a trusted visitor, unlock its own door, and unknowingly invite one of nature's greatest master infiltrators inside. Chapter 2. Finding the perfect victim. No master thief begins by choosing a random house. No expert spy walks through the first unlocked door, and no successful virus drifts through the body hoping that any cell will do. That is perhaps the first illusion we must abandon. Viruses do not attack at random. They search, patiently, relentlessly, with astonishing precision. If Chapter 1 revealed that a virus is little more than genetic instructions wrapped inside a microscopic shell, Chapter 2 asks a more unsettling question. How does something that cannot see, cannot think, cannot smell, and cannot make decisions consistently find exactly the right cell among nearly 37 trillion possibilities? At first, this seems impossible. Imagine releasing a single key into a city containing trillions of doors. The odds of finding the correct lock should be essentially zero. Yet viruses accomplish this feat every single day. The answer lies not in intelligence, but in chemistry. Long before detectives began solving crimes with fingerprints, nature had already invented an even more precise identification system. Every living cell carries thousands of proteins on its surface. Some receive hormones, some exchange nutrients, others allow neighboring cells to communicate. These proteins are known as receptors. You can think of them as door handles, identification badges, communication antennas, or docking stations. Without them, cells could not coordinate the countless activities required for life. But every useful system creates an opportunity for exploitation. Somewhere in the distant history of evolution, viruses discovered that if they could imitate the molecules these receptors normally recognize, they would never need to break into a cell. The cell would invite them inside. the lock would open itself. Consider one of the most discussed receptors in modern medicine, ACE2. Before 2020, relatively few people outside scientific circles had ever heard its name. Today, it is recognized around the world. Yet ACE2 never evolved for viruses. Its true purpose is remarkably ordinary. It helps regulate blood pressure, inflammation, and the balance of fluids throughout the body. It is an important component of healthy human physiology. The coronavirus simply learned to exploit it. Its famous spike proteins fit the ACE2 receptor with extraordinary precision, much like a counterfeit key crafted by an expert locksmith. The receptor believes it is interacting with a familiar partner. Instead, it has unknowingly welcomed an intruder. But ACE2 is only one lock among countless others. HIV prefers an entirely different entrance. Rather than targeting cells lining the airways, HIV seeks out one of the most important defenders of the immune system, the CD4 receptor. CD4 appears on helper T cells, white blood cells responsible for coordinating immune responses throughout the body. They function like military commanders, directing other immune cells where to fight and when to attack. Without them, organized defense begins to collapse. Yet even CD4 alone is not enough. HIV demands a second lock, a co-receptor. Most commonly, this is CCR5. Only when both CD4 and CCR5 are present can many strains of HIV successfully enter the cell. It is as though a bank vault requires not one security code, but two entered simultaneously. This remarkable requirement explains one of the most fascinating discoveries in human genetics. A small percentage of people inherit a mutation known as CCR5 del32. Their CCR5 receptors are missing or severely altered. For many forms of HIV, the second lock simply does not exist. Without that lock, the door never opens. The virus arrives, searches, touches the surface, and leaves empty-handed. The victim was never truly vulnerable. Other viruses employ entirely different strategies. Many cold-causing viruses recognize a receptor known as ICAM1, or intercellular adhesion molecule 1. Normally, ICAM1 helps immune cells communicate and migrate through tissues during inflammation. Once again, the receptor evolved to support life. The virus merely repurposed it for invasion. Then there is heparan sulfate. Unlike highly specialized receptors found only on particular cells, heparan sulfate is astonishingly widespread. It coats the surfaces of many different cell types throughout the body. Some viruses use it as an initial landing pad, a place to slow down and attach before locating a more specific receptor that grants true entry. Imagine an aircraft first reaching the airport before taxiing toward the correct gate. Attachment alone is not enough, but it is an essential first step. These receptor preferences reveal an extraordinary truth. Viruses are surprisingly selective. Influenza viruses rarely wander into nerve cells, not because they dislike them, but because the appropriate receptors are largely absent. Instead, influenza targets cells lining the respiratory tract. There, specialized sugar molecules called sialic acids provide exactly the molecular landscape influenza requires. The nose, the throat, the windpipe, the lungs. These become its preferred territory, not through choice, through compatibility. HIV displays an even narrower preference. It does not attack muscle cells, it ignores skin cells. It bypasses most organs entirely. Instead, it relentlessly searches for immune cells carrying CD4 and appropriate co-receptors. Its battlefield is not the lungs. It is the immune system itself. Hepatitis viruses tell another story. Many display a remarkable attraction for liver cells. The liver performs hundreds of biochemical functions essential for survival. It filters blood, processes nutrients, produces proteins, stores energy. Certain hepatitis viruses have evolved precisely to recognize receptors concentrated on these remarkable cells. The result is an infection that largely remains within one organ while sparing many others. Then comes rabies. Perhaps no viral journey is stranger. Rather than multiplying primarily in the lungs or bloodstream, rabies seeks neurons, nerve cells. Its destination is not merely tissue. It is the nervous system itself. Once inside peripheral nerves, the virus begins an astonishing migration toward the brain, traveling along neuronal pathways that function almost like microscopic highways. Its route appears deliberate, its destination terrifyingly precise. Every one of these examples demonstrates a fundamental principle in virology. Scientists call it cell tropism. Cell tropism simply means that every virus displays a preference for particular cell types. Not every cell is suitable. Not every lock fits. Only certain cells provide the receptors, internal machinery, and biological environment required for successful infection. But the mystery deepens. Viruses are selective not only about cells, they are selective about species. This concept is known as host range. Some viruses infect only one species throughout their entire existence. Others move comfortably among many. Bacteriophages infect bacteria but ignore humans completely. Countless plant viruses devastate crops while posing no threat whatsoever to animals. Likewise, many animal viruses spend millions of years circulating within bats, rodents, birds, or primates without ever successfully entering the human population. Why? Because possessing the wrong receptors is rather like possessing the wrong key. The virus may arrive, it may touch the surface, it may even remain attached briefly. But if the molecular fit is imperfect, nothing follows. The lock refuses to turn. This invisible boundary is known as the species barrier. For countless viruses, it is nearly impossible to cross. A virus adapted to birds often recognizes bird receptors far better than human ones. A virus specialized for bats may find human cells completely incompatible. The barrier protects us every single day in ways we never notice. Every moment, humans encounter enormous numbers of viruses that simply cannot infect us, not because our immune systems defeat them, but because they never gain entry in the first place. Yet history reminds us that barriers are not walls, they are filters. Occasionally, a mutation appears, a tiny alteration affecting the proteins covering a virus. Perhaps only a single amino acid changes, perhaps two. Suddenly, the viral key fits a new lock just well enough. Not perfectly, but sufficiently. The virus enters. It replicates. Natural selection favors this new ability. Generation after generation, the fit improves. Eventually, a virus once confined to another species discovers humanity. Many of history's most significant outbreaks began with precisely such events. A crossing. A mismatch becoming a match. A locked door quietly opening for the very first time. The detective investigating this mystery might therefore reach an unexpected conclusion. The virus is rarely the first actor in the story. The receptor is. Without the receptor, there is no attachment. Without attachment, there is no entry. Without entry, there is no infection. The entire chain of events depends upon an interaction measured in billionths of a meter. One molecular handshake, one perfect fit, one lock recognizing what appears to be the correct key, which raises an unsettling thought. Perhaps the virus does not truly deceive the cell. Perhaps the cell is simply performing the task evolution assigned to it. Receiving signals, welcoming trusted molecules, maintaining normal life. The tragedy is that the virus has learned to imitate those trusted visitors with extraordinary skill. Every successful infiltration depends upon that deception. Every epidemic begins with countless individual cells making what appears to be an entirely ordinary decision. They recognize a familiar shape. They engage a familiar receptor. They initiate a familiar biological process. Only afterward does the catastrophe begin. And that realization brings us to the next stage of our investigation. Because finding the correct victim is only the beginning. A key that fits the lock has accomplished very little. The door must still open. The virus must still cross the threshold. The cell must somehow transform from cautious guardian into willing accomplice. For perhaps the greatest secret of all is this. The cell is not stolen by force. It opens the door itself, breaking into the cell. Every great heist follows the same unwritten rule. The best thief is never the one who breaks the strongest vault. The best thief is the one who convinces the owner to unlock it. That is precisely why viruses have survived for hundreds of millions of years. They are not battering rams. They are master impersonators. By the time a virus enters a cell, the hardest part of its mission has already been completed. The correct victim has been identified. The proper receptor has been found. The molecular handshake has taken place. Now comes the second phase of the operation, crossing the border. Imagine standing outside one of the most heavily protected buildings ever constructed. Its walls constantly repair themselves. Its security cameras never sleep. Its guards communicate instantly. Its entrances are monitored every second. Breaking through the wall would be almost impossible. Fortunately for the intruder, there is no need. The employees are perfectly capable of opening the doors themselves. Living cells cannot remain sealed off from the outside world. If they did, life would end within minutes. Nutrients must enter. Hormones must deliver instructions. Waste products must leave. Immune cells must communicate. The membrane surrounding every cell is not a fortress. It is more like the entrance to a busy international airport. Thousands of authorized arrivals occur every second. The virus simply learns to arrive disguised as an invited guest. Once the viral proteins bind to the correct receptor, something remarkable happens. The cell itself begins changing. Proteins beneath the membrane reorganize. The flexible surface starts bending inward. The entrance process has already begun. No alarms, no explosions, no shattered walls. The victim is unknowingly participating in its own invasion. Virologists have identified several major strategies used by viruses to cross the cellular border. Each one resembles a different style of carefully planned burglary. The first is perhaps the most elegant. Fusion. Imagine two soap bubbles floating toward one another. When they touch under the right conditions, they become one larger bubble. Their membranes merge. Their contents intermingle. Certain viruses accomplish something astonishingly similar. Instead of punching through the cell membrane, they simply blend with it. Their outer lipid envelope fuses directly with the membrane surrounding the cell. For a brief moment, two separate boundaries become one continuous surface. A doorway appears where none existed before. The viral contents quietly slip into the cell's interior. The virus itself almost disappears during the process. HIV uses this remarkable strategy. Once its envelope proteins engage CD4 and the necessary co-receptors discussed in our previous chapter, molecular machinery changes shape dramatically. The viral envelope fuses with the host membrane. Its genetic cargo enters the cell almost silently. No broken walls, no forced entry, simply two membranes becoming one. Certain coronaviruses, including SARS-CoV-2, can also employ membrane fusion after activation by enzymes located on the host cell surface. These enzymes act almost like security personnel, unknowingly validating a forged passport. Only after receiving this unexpected assistance can fusion proceed efficiently. The cell believes it is completing an ordinary biological task. Instead, it has opened the first gate. Not every virus chooses this route. Many rely on a strategy called endocytosis. If fusion resembles quietly walking through the front entrance, endocytosis resembles being escorted inside in a delivery package. Cells perform endocytosis constantly. It is one of their most important housekeeping functions. Small portions of the membrane fold inward, forming tiny bubbles called vesicles. These membrane-bound containers transport nutrients, hormones, and signaling molecules into the cell. The process is entirely normal, entirely necessary. Viruses merely exploit it. Picture a bank employee accepting a sealed package from someone wearing an official uniform. The package passes every inspection. It is logged into the system. It is carried directly into the building. Only later does anyone discover what was actually inside. Influenza viruses frequently depend upon endocytosis. After attaching to receptors decorated with sialic acid molecules lining the respiratory tract, the cell responds exactly as it would to many legitimate molecular visitors. The membrane wraps around the virus. A vesicle forms. The virus is brought safely inside. Everything has proceeded according to the cell's own operating manual, except the cargo was never supposed to be there. Then comes an even stranger strategy, macropinocytosis. The name sounds intimidating. The concept is surprisingly familiar. Imagine someone convincing a homeowner to bring an enormous package into the living room before opening it. The package is far larger than an ordinary parcel, yet the homeowner willingly carries it inside. Cells occasionally perform something remarkably similar. Rather than importing tiny individual molecules, they engulf large volumes of surrounding fluid. It is almost as though the membrane reaches outward, scoops up everything nearby, and draws it inward within a gigantic bubble. Some viruses have evolved to trigger precisely this behavior. Instead of sneaking through a doorway, they persuade the entire doorway to wrap itself around them. Large viruses, including certain emerging pathogens, exploit this process to gain entry into cells that would otherwise be difficult to penetrate. The cell performs the heavy lifting. The virus merely takes advantage of the invitation. Another strategy appears almost primitive by comparison: direct penetration. Unlike enveloped viruses that merge membranes, some non-enveloped viruses interact with the cell surface before creating tiny openings through which their genetic material enters. Adenoviruses provide an excellent example. Their sturdy protein shells do not possess delicate lipid envelopes capable of fusion. Instead, after attachment and internalization, remarkable structural changes occur. Mechanical forces, acidity, and host proteins gradually weaken the viral shell. Eventually, the virus escapes from intracellular compartments into the cytoplasm. It is less dramatic than breaking down a wall, more like quietly slipping through a service corridor that no one remembered existed. Despite these different methods, every successful virus shares one astonishing characteristic. None truly forces its way inside. The cell performs nearly every step. It bends its own membrane, forms its own vesicles, moves the incoming particle using its own molecular motors, consumes its own energy. From beginning to end, the victim unknowingly assists the criminal. The detective examining this case would discover something deeply unsettling. There are almost no signs of violence, no shattered locks, no damaged entrances, no emergency alarms, only normal cellular behavior redirected toward extraordinary consequences. But crossing the membrane is not the final objective. It is merely the beginning. Imagine robbers entering a bank while still wearing oversized disguises that prevent them from moving freely. They cannot access computers. They cannot open vaults. They cannot even reach the money. First, the disguises must come off. Viruses face the same problem. Their genetic instructions remain locked inside protective protein shells. Those shells were essential during the dangerous journey outside the cell. Inside, they become obstacles. This next stage carries a wonderfully descriptive name: uncoding. The word sounds almost gentle, yet it marks one of the most decisive moments in the entire infection. The capsid begins to fall apart. Sometimes the acidic environment inside cellular compartments weakens its structure. Sometimes host enzymes gradually dismantle it. Sometimes internal pressure causes it to burst open. Every virus has evolved its own carefully timed solution. The result, however, remains the same. The protective shell disappears. The genetic material hidden within is finally exposed. Imagine opening a sealed envelope after months of searching for it. The paper inside contains detailed instructions. Those instructions cannot act alone. But once someone begins reading them, events unfold rapidly. The viral genome now enters the cell's internal world. Whether composed of DNA or RNA, it carries a complete set of molecular orders. Not enough information to build an entire organism, but enough to transform another organism into a factory. Curiously, the virus itself almost seems to vanish. The magnificent spikes, the elegant geometric capsid, the recognizable external appearance seen in countless scientific illustrations, all begin disappearing from the story. Investigators searching the infected cell may no longer find an intact viral particle. Instead, only fragments remain, pieces of protein, scattered components, and somewhere inside, genetic instructions now blending into the busy molecular environment of the host. The intruder has dissolved into the crowd. This is perhaps the cleverest moment of the entire operation. The visible criminal disappears. The invisible instructions remain. From this point forward, the infection is no longer driven by the virus's exterior. Everything depends upon information, genetic information, encoded instructions, a biological script waiting to be read by machinery that never evolved to serve it. The cell still appears remarkably ordinary. Its membrane remains intact. Its mitochondria continue generating energy. Its ribosomes continue assembling proteins. Its nucleus continues managing countless daily responsibilities. Nothing about the cell announces that a silent takeover has already begun, which makes the next question all the more disturbing. If the virus has effectively disappeared, who exactly is now giving the orders? Because in the next chapter, we will witness one of the greatest acts of biological deception ever discovered. The virus will not build a single protein with its own machinery. It has none. Instead, it will persuade the cell's own factories to manufacture an invading army. And perhaps the most dangerous thing about a virus is this: once it gets inside, it almost disappears. Chapter 4. The greatest biological hack ever invented. A successful bank robbery does not end when the thieves enter the building. Entering is only the beginning. The true objective is much more ambitious. Control the security system, control the communication network, control the employees. If every worker inside the bank continues doing their job, but unknowingly begins serving the robbers instead of the owners, the robbery has become something far more dangerous than theft. It has become a takeover. That is exactly what happens inside an infected cell. By the end of our previous chapter, the virus had accomplished the impossible. It had crossed the membrane, its protective shell had disappeared, its genetic instructions had been released. But those instructions, remarkable as they are, still possess one enormous limitation. They cannot build anything by themselves. A strand of RNA cannot assemble proteins. A strand of DNA cannot manufacture enzymes. Genetic information is merely information. It is a recipe, and a recipe cannot cook itself. Somewhere inside every living cell stands one of biology's most extraordinary inventions, the ribosome. If DNA is the library and genes are the books, then ribosomes are the master craftsmen capable of turning written instructions into physical reality. Everything your body has ever built has depended upon them. Every muscle fiber, every antibody, every hormone, every enzyme, every receptor discussed in our previous chapters. None of them exist until ribosomes assemble them one amino acid at a time. Imagine entering the largest factory ever constructed. Thousands of assembly lines stretch toward the horizon. Robotic arms never pause. Conveyor belts move continuously. Workers require no sleep, no vacations, no weekends. 24 hours a day, every second of your life, these microscopic factories manufacture the proteins that keep you alive. The astonishing truth is that ribosomes do not understand biology. They do not know whether they are building something beneficial or something catastrophic. They possess no judgment, no loyalty, no awareness. They simply read instructions, and then they obey. To appreciate the brilliance of the viral strategy, we first need to understand how those instructions normally arrive. Deep inside the nucleus of a healthy cell rests its DNA, protected, carefully organized, too valuable to expose directly to the crowded environment of the cytoplasm. Instead of sending DNA itself, the cell creates temporary working copies. These copies are called messenger RNA, or simply mRNA. Messenger RNA functions remarkably like a printed blueprint delivered from an architect's office to a construction site. The original plans remain safely archived. The workers receive only the copy they need. When a protein is required, the cell produces the appropriate mRNA. The messenger travels outward. Eventually, it encounters a ribosome. The ribosome locks onto the message, begins reading 3 genetic letters at a time. Each 3-letter code specifies one amino acid. One amino acid joins another, then another. Gradually, an entirely new protein begins to emerge. Scientists call this remarkable process translation. The name is beautifully appropriate because the ribosome is literally translating genetic language into physical matter. Life itself depends upon this continuous translation. Now imagine someone secretly replacing the blueprints arriving at every construction site throughout an entire city. The workers remain exactly the same. The machinery remains exactly the same. The electricity continues flowing. The factories never stop operating. Only the instructions have changed. Without realizing it, the workers begin constructing an entirely different project. That is the virus's masterpiece. Most viruses never attempt to build their own ribosomes. Why should they? Constructing such sophisticated molecular machines would require enormous amounts of genetic information. Instead, evolution discovered something infinitely more efficient. Simply hijack the existing factories. Once viral genetic material reaches the cytoplasm, or in some cases the nucleus before returning as messenger RNA, the infected cell unknowingly begins producing viral mRNA. To the ribosome, these messages appear surprisingly ordinary. The ribosome does not recognize nationality. It does not ask where the instructions originated. It does not pause to verify ownership. It simply reads whatever messenger RNA arrives. Human message, viral message. To the ribosome, they are merely sequences waiting to be translated. This simple fact may represent one of the greatest vulnerabilities in all of biology. The cell built perfect factories. The virus learned how to submit counterfeit work orders. Naturally, cells are not entirely defenseless. Over billions of years, they evolved ways to prioritize their own messenger RNA and regulate protein production with extraordinary precision. Viruses, however, evolved equally ingenious methods for bypassing those safeguards. One of the most fascinating strategies carries a dramatic name: cap snatching. At first hearing, it almost sounds like something from a detective novel. The term is surprisingly literal. Most human messenger RNAs possess a protective chemical structure attached to one end. Known as the 5' cap, this tiny molecular modification helps stabilize the message and allows ribosomes to recognize it efficiently. Influenza viruses discovered a remarkable shortcut. Instead of manufacturing these caps themselves, they steal them. Inside infected cells, influenza enzymes slice the capped beginnings from normal cellular messenger RNAs. These stolen fragments are then attached to viral RNA. The result is astonishing. The virus disguises its own instructions using genuine cellular identification. To the ribosome, the counterfeit document appears authentic. The forged passport passes inspection. The factory begins production. Meanwhile, the cell's own messages have been damaged or destroyed. Production gradually shifts toward viral priorities. It is identity theft on a molecular scale. Other viruses employ an entirely different strategy. Instead of stealing identification, they eliminate the need for it. Scientists call these remarkable structures internal ribosome entry sites, or simply IRIS. Normally, ribosomes begin reading messenger RNA by recognizing the protective cap at one end. IRS changes the rules completely. It acts almost like a hidden emergency entrance built directly into the genetic instructions themselves. Rather than approaching through the usual front gate, the ribosome is recruited from within. Imagine a hacker discovering a forgotten maintenance tunnel beneath a heavily guarded factory. Security at the main entrance remains excellent. It no longer matters. The intruder never uses the front door. Viruses such as poliovirus and hepatitis C virus have perfected this astonishing technique. Even when normal protein production begins shutting down, IRS-containing viral messages continue recruiting ribosomes with remarkable efficiency. Then comes perhaps the boldest strategy of all: host shutoff. The phrase sounds technical. Its consequences are devastating. Instead of merely increasing production of viral proteins, many viruses actively suppress production of the cell's own proteins. Messenger RNAs are destroyed. Translation factors become disabled. Protein synthesis machinery is redirected. Some viruses accelerate the degradation of host messenger RNA. Others block its export from the nucleus. Still others interfere directly with ribosomal function. The effect resembles a hostile corporate takeover. Imagine entering a vast manufacturing complex. One by one, every employee producing legitimate products is dismissed. Their assembly lines fall silent. The machinery is not destroyed. It is reassigned. By the end of the day, every conveyor belt manufactures only the invader's products. The factory remains astonishingly productive. It simply no longer serves its original owner. SARS-CoV-2 employs particularly sophisticated methods to interfere with normal host protein production. Certain viral proteins reduce the cell's ability to translate many of its own messenger RNAs while allowing viral messages to continue dominating the machinery. The exact mechanisms remain an active field of research, but the overall objective is unmistakable: silence the host, amplify the virus, produce more copies, produce them faster, produce almost nothing else. Perhaps the most extraordinary aspect of this biological takeover is its invisibility. Nothing appears unusual from outside the cell. Its membrane remains intact. Its organelles remain present. Energy production continues. The ribosomes never stop working. If anything, many infected cells become extraordinarily busy. Thousands upon thousands of proteins emerge every minute. The machinery has not failed. It has become astonishingly efficient. Only one detail has changed. The products leaving the assembly lines no longer belong to the cell. Imagine interviewing every worker inside this microscopic factory. None would report a crisis. Every machine functions normally. Every conveyor belt operates flawlessly. Every robotic arm performs exactly as designed. Yet collectively, they are constructing an army: spike proteins, capsid proteins, polymerases, proteases, structural components, everything required to build entirely new viruses. The workers never notice. The factory never protests. The blueprints look legitimate. The instructions were followed. The consequences were catastrophic. From the perspective of the virus, this may be the greatest biological hack ever invented. It never built its own manufacturing plants. It never evolved independent protein factories. It simply discovered how to persuade another organism to provide everything. The host supplies the energy, the amino acids, the ribosomes, the quality control systems, even the waste disposal. The virus contributes little more than information, yet information, properly delivered, can redirect an entire civilization. Perhaps that is the deeper lesson hidden within every infected cell. Power does not always belong to the strongest machine. Sometimes it belongs to whoever controls the instructions. And now the virus possesses those instructions. The factory is still humming. The conveyor belts are still moving. The workers have never been busier. But something fundamental has changed. The cell has not died. It has not stopped functioning. It has simply forgotten who it is working for. Which brings us to the next stage of our investigation. The viral proteins are now complete. The molecular workers have assembled every necessary component. Only one task remains: construction, assembly, replication on a scale almost beyond imagination. Because in the next chapter, we will discover how a single microscopic intruder transforms one healthy cell into a factory capable of producing thousands, sometimes tens of thousands, of new viruses before anyone realizes what has happened. For perhaps the most chilling truth of all is this: the cell never stopped working. It simply stopped working for itself. Chapter 5: Turning a Living Cell into a Virus Factory. Every great conspiracy reaches a moment when secrecy is no longer enough. The infiltrator has entered. The guards have been deceived. The communication network has been compromised. Now comes the true objective: production. Until this point in our investigation, the virus has behaved like a master spy. It located the correct victim. It persuaded the cell to unlock its own door. It disappeared into the crowded interior. It quietly redirected the ribosomes, convincing them to manufacture viral proteins instead of the cell's own. But proteins alone are not a virus. A pile of bricks is not a house. A collection of gears is not a clock. To create new viruses, something even more remarkable must happen. The genetic instructions themselves must be copied. Again and again and again. Imagine discovering a secret printing press hidden beneath an ordinary factory. Every few minutes, another complete set of classified blueprints rolls off the machine. Soon there are dozens, then hundreds, then thousands. Every copy is capable of building another factory somewhere else. This is the moment when one invader becomes an army. The process begins with a deceptively simple question: how does a virus copy its own genome when it arrived carrying almost nothing? The answer depends entirely upon what kind of virus has entered the cell. Not all viruses speak the same genetic language. Some carry DNA, others carry RNA. A few perform tricks so extraordinary that they seem to violate the usual rules of biology. Let us begin with the DNA viruses. These viruses possess genomes made from the same molecule that stores the genetic information inside your own cells. Because the language is familiar, many DNA viruses take advantage of the host cell's existing machinery. Some travel all the way into the nucleus, the secure archive where the cell stores its own chromosomes. There, surrounded by enzymes evolved to duplicate human DNA, the viral genome quietly joins the queue. The cell believes it is copying another piece of DNA. Instead, it is unknowingly producing viral genomes. Some DNA viruses, however, bring specialized proteins of their own to assist with the process. Large viruses cannot always depend entirely upon the host. They supplement the machinery. They optimize it. They ensure that replication proceeds according to their own timetable rather than the cell's. Now consider RNA viruses. Here the mystery becomes far more intriguing. Human cells do not normally copy RNA into new RNA. They have no standard enzyme dedicated to that task. So RNA viruses solve the problem themselves. Hidden within many RNA viruses is an extraordinary molecular machine called RNA-dependent RNA polymerase. Its name is technical. Its purpose is elegantly simple. It reads one RNA strand and produces another, over and over again. Without this enzyme, many RNA viruses would reach a dead end. Their instructions could never multiply. Instead, RNA polymerase transforms a single viral genome into dozens, hundreds, or thousands of new copies. Each one represents another complete instruction manual waiting to be packaged. Then we encounter perhaps the greatest rebel in molecular biology: retroviruses. Most living organisms follow a familiar flow of information. DNA becomes RNA. RNA becomes protein. For decades, scientists considered this one-way traffic. Then retroviruses revealed an astonishing exception. Viruses such as HIV carry RNA rather than DNA. Yet after entering a cell, they perform something that once seemed impossible. Using an enzyme known as reverse transcriptase, they rewrite their RNA into DNA. The direction has been reversed. The messenger becomes the master copy. Once converted into DNA, the viral genome slips into the host chromosomes. It becomes part of the cell's own genetic archive. From that moment onward, every time the cell reads its own DNA, it may unknowingly read viral instructions as well. The infiltrator has not merely occupied the building, it has rewritten the building's blueprints. Reverse transcriptase deserves special attention for another reason. Unlike many enzymes used by our own cells, it is astonishingly inaccurate. It copies quickly, but not carefully. Every new genome carries the possibility of mistakes. Those mistakes, as we shall soon discover, may become one of evolution's most powerful engines. Regardless of whether the virus carries DNA or RNA, replication rarely occurs in isolation. Inside infected cells, viral proteins gather together with host proteins to create temporary molecular workshops. Scientists call these remarkable structures replication complexes. Imagine abandoned warehouses suddenly transformed into secret assembly plants. The walls remain the same, the roof remains the same, but inside, every table, every machine, and every worker has been reorganized around a single objective: build more viruses. Within these hidden factories, newly copied genomes accumulate while ribosomes continue producing viral proteins, capsid proteins, envelope proteins, spike proteins, polymerases, proteases, structural enzymes, accessory proteins. Every component required to construct another complete viral particle gradually appears. The assembly lines never rest. The speed can be astonishing. One virus becomes 10, 10 become 100, 100 become 1,000, sometimes far more. Exactly how many new viruses emerge depends upon the species involved, the type of infected cell, and the conditions inside the host. Some viruses produce only modest numbers before the cell dies. Others generate tens of thousands of descendants from a single infected cell. Imagine a counterfeit printing operation so efficient that one forged banknote becomes 100, then 1,000, then entire truckloads before anyone notices the original is missing. This explosive multiplication explains why viral infections often appear to accelerate so suddenly. During the earliest stages, only a handful of cells may be infected. Each quietly becomes a factory. Each factory manufactures hundreds or thousands of new particles. Those particles spread outward, converting neighboring cells into additional factories. The expansion is no longer linear. It becomes exponential. 1, 10, hundreds, thousands, millions, billions. What seemed like an invisible whisper rapidly becomes a biological storm. Yet hidden inside this astonishing productivity lies a flaw, or perhaps a secret weapon. No copying process is perfect. Even the remarkable enzymes inside human cells occasionally make mistakes. Fortunately, our cells possess sophisticated proofreading systems capable of correcting many errors before they become permanent. Many viruses enjoy no such luxury. RNA polymerases often copy genomes with remarkable speed but relatively poor accuracy. Reverse transcriptase is even more error-prone. Letters change, small sections disappear, others are inserted. Most of these mutations accomplish nothing useful. Some damage the virus so severely that it can no longer reproduce. Those defective copies simply vanish from the evolutionary story. But every so often, an accident becomes an advantage. Perhaps a mutated spike protein binds receptors more effectively. Perhaps an altered enzyme replicates faster. Perhaps a slight structural change helps the virus evade antibodies. The mutation survives. Future generations inherit it. Natural selection quietly performs its work. What began as an error becomes an innovation. This is evolution unfolding in real time. Unlike humans, whose generations require decades, many viruses complete entire reproductive cycles within hours. Evolution that might require thousands of years in larger organisms can unfold astonishingly quickly among viruses. Each infected individual becomes not merely a patient, but an evolutionary laboratory. Every infected cell produces enormous numbers of genomes. Every copied genome presents another opportunity for mutation. Every mutation becomes another experiment. Most experiments fail. A tiny number succeed. Those rare successes gradually reshape entire viral populations. This is how variants emerge, not because viruses consciously adapt, not because they plan, but because countless imperfect copies continuously explore new genetic possibilities. Imagine a master codebreaker trying millions of combinations against a locked safe. Almost every attempt fails. Eventually, one works. Nature follows a similar principle. The virus keeps making copies. The copies keep changing. The environment quietly selects whichever versions happen to survive most effectively. The detective following our mystery might finally realize something profound. The virus does not merely invade, it improvises. Every infection writes a slightly different chapter. Every host becomes another testing ground. Every copied genome asks the same silent question. Could this version survive better than the last? Sometimes the answer is no. Occasionally, history changes. This realization transformed modern virology. Scientists no longer view viruses as static objects. They are moving populations, clouds of related genomes, families of slightly different descendants competing continuously within the same host. The virus infecting one person today may already differ subtly from the one infecting another tomorrow. Evolution never pauses. Even while the infected individual sleeps, countless molecular experiments continue unfolding inside their cells. Which brings us to an unsettling realization. The virus has now achieved nearly everything it came to accomplish. The cell's factories are fully operational. Its genomes have been copied, its proteins assembled, its descendants are beginning to take shape. But the body is not entirely defenseless. Somewhere beyond these microscopic factories, silent sentinels have begun noticing unusual activity. Signals are spreading, warnings are being transmitted, an entirely different army is preparing its response. Because in our next chapter, the investigation shifts away from the virus and toward the remarkable intelligence of the infected cell itself. For although the factory has been hijacked, someone inside may already have realized that the blueprints have changed. And perhaps the most astonishing truth of all is this: every time a virus copies itself, it is also quietly testing a new version of itself. Chapter 6: The Invisible War Inside the Cell. The perfect crime lasts only until someone notices that something is wrong. For several chapters, our invisible intruder has seemed almost unstoppable. It found the correct victim. It persuaded the cell to unlock its own door. It hijacked the ribosomes. It transformed an ordinary cell into a factory producing hundreds, sometimes thousands of new viral particles. To an outside observer, the operation appeared flawless. No shattered membrane, no broken machinery, no dramatic explosion, only a quiet change of ownership. If this were the entire story, viruses would have conquered life long ago. But every great mystery contains an investigator. Every hidden conspiracy eventually encounters someone who begins asking uncomfortable questions. Inside every living cell, there is already a detective. It never sleeps. It never grows bored. And unlike human investigators, it has been solving the same crime for more than 1 billion years. The detective has no eyes, no brain, no consciousness, yet it recognizes one thing with astonishing accuracy: the difference between self and something that should not be here. This ancient security system is known as innate immunity. Long before humans evolved antibodies, long before specialized T cells or memory B cells existed, long before mammals walked across the earth, cells had already developed a primitive but remarkably effective alarm system. Imagine a medieval city surrounded by thick stone walls. The guards do not know the identity of every possible enemy. They cannot recognize every invading army. Instead, they watch for familiar signs: smoke rising where there should be none, footprints outside the gates, broken signals from distant watchtowers. They are not searching for individuals. They are searching for patterns. Cells do exactly the same thing. Rather than identifying every virus one by one, they search for molecular fingerprints shared by many invaders. Scientists call these fingerprints pathogen-associated molecular patterns, or PAMPs, pieces of double-stranded RNA, unusual viral RNA structures, DNA appearing where it should never exist, chemical signatures rarely found in healthy human cells. The cell does not need to know which virus has arrived. It simply needs to recognize that something profoundly abnormal is happening. To detect these warning signs, cells employ an extraordinary network of molecular sentinels known as pattern recognition receptors, or PRRs. Think of them as detectives stationed throughout an enormous city. Some patrol the gates, others monitor the streets. Still others inspect activity deep inside government buildings. Together, they form one of biology's oldest intelligence networks. Among the first investigators to encounter an invading virus are the toll-like receptors, usually abbreviated as TLRs. The name itself carries a fascinating history. The first toll gene was discovered not in humans, but in fruit flies, where it played a role in embryonic development before scientists realized its importance in immunity. Today, toll-like receptors are recognized as some of the immune system's earliest warning devices. Different TLRs specialize in different clues. Some search for viral RNA trapped inside tiny membrane compartments called endosomes. Others recognize unusual DNA molecules. Each receptor functions like a detective trained to recognize one particular style of criminal behavior. The moment the evidence appears, the investigation begins. But viruses rarely remain confined to the cell's outer regions. Sooner or later, many release their genomes into the cytoplasm. There, another team of investigators takes over. Meet Rig-I and its close partner, MDA5. Unlike toll-like receptors positioned near entry points, these proteins patrol the interior itself. Imagine undercover investigators walking quietly among factory workers, inspecting every document that passes through the building. Most messenger RNA looks perfectly ordinary. It bears familiar chemical modifications, expected structures, recognizable signatures. Viral RNA often does not. It contains unusual ends, unexpected double-stranded regions, patterns rarely produced by healthy human cells. The moment Rig-I encounters one of these suspicious molecules, it changes shape dramatically. The evidence has been found. The alarm cannot remain silent any longer. MDA5 performs a similar duty, although it specializes in recognizing longer stretches of viral double-stranded RNA produced during replication. Together, Reg-I and MDA5 provide overlapping surveillance, ensuring that few viral genomes escape unnoticed. At least, that is the plan. Because recognition alone accomplishes nothing. Finding the criminal is only the first step. The information must reach headquarters. Inside the infected cell, molecular signals begin racing through complex biochemical pathways. Proteins activate one another like falling dominoes. One switch flips another. That switch activates several more. Within minutes, dozens of genes previously lying dormant suddenly awaken. The cell has reached a terrifying conclusion. It has been compromised. Now comes perhaps the most remarkable act of selflessness in cellular biology. The infected cell knows it may not survive, but it can still warn its neighbors. Its most urgent message is carried by a family of proteins called interferons. The name is beautifully descriptive. They interfere, not with healthy cells, but with viral replication. Imagine a town discovering enemy soldiers hidden within one neighborhood. The local authorities know they cannot save the occupied buildings. Instead, they send urgent radio broadcasts across the entire region. Lock your doors, prepare your defenses, destroy sensitive documents, expect infiltration. Interferons perform exactly this function. Released by infected cells, they diffuse outward into surrounding tissues. Neighboring cells receive the warning almost immediately. Although these cells remain uninfected, they begin preparing for battle. Hundreds of defensive genes activate. Protein production changes. Antiviral molecules accumulate. Doors begin closing before the enemy even arrives. The neighborhood transforms into hostile territory for the virus. Perhaps the most extraordinary aspect of interferons is that they represent communication rather than combat. The infected cell sacrifices precious time and energy to protect others. It sends an SOS, a microscopic distress signal. One cell may fall. The tissue survives. But warnings alone cannot stop an active infection. The receiving cells must also deploy weapons. One of the first to appear is PKR, short for protein kinase R. PKR behaves almost like an emergency circuit breaker inside the factory. Normally, ribosomes continue manufacturing proteins around the clock. When PKR detects viral double-stranded RNA, it activates rapidly. Its target is one of the key molecules required for protein synthesis. PKR modifies this molecule chemically, bringing much of the cell's translation machinery to a halt. Imagine investigators discovering counterfeit blueprints flowing through a factory. Rather than allowing production to continue, they pull the master power switch. Everything slows. The virus loses access to the assembly lines it desperately needs. The cell suffers as well, but survival sometimes requires sacrifice. Another remarkable defense follows, the OAS system, short for oligoanylate synthetase. The name sounds intimidating. Its strategy is elegantly simple. When activated by viral RNA, OAS manufactures tiny signaling molecules that awaken another molecular weapon, RNase L. If PKR shuts down the factory, RNase L begins shredding documents, messenger RNAs, viral RNAs, even some cellular RNAs. Anything that could support viral replication becomes a target. Imagine investigators realizing that secret plans have been stolen. Rather than allowing the enemy to continue reading them, every remaining copy is fed directly into industrial shredders. Information disappears. Without instructions, production collapses. The virus finds itself trapped inside a factory that has intentionally destroyed its own blueprints. It is a desperate strategy, but remarkably effective. By now, our investigation has revealed something profound. Cells are not passive victims. They fight, they communicate, they warn one another. They willingly reduce their own productivity to slow the invader's progress. They behave less like isolated biological units and more like citizens defending a civilization. The virus enters one building, the entire city responds. And yet, history reminds us that not every warning reaches its destination. Not every emergency broadcast is transmitted. Not every alarm succeeds. Throughout evolution, viruses have studied these defensive systems with extraordinary patience. Every interferon signal, every receptor, every molecular investigator, every emergency response. They have learned where the weaknesses lie. Some interrupt communication, others silence alarms before neighboring cells can hear them. Some hide the evidence entirely, ensuring that detectives never recognize the crime in the first place. Our detective story, it seems, has only become more complicated because finding the criminal is one challenge. Preventing the criminal from cutting the telephone lines is another entirely. The invisible war unfolding inside every infected cell is not simply a contest of strength. It is a battle of information. Detection against deception. Communication against silence. Warning against concealment. Every second, billions of microscopic decisions determine whether an infection expands or quietly ends before anyone ever notices. Which brings us to perhaps the most unsettling question in our entire investigation. What happens if the emergency signal is never sent? What if the detectives never discover the evidence? What if the city continues sleeping peacefully while the invader quietly captures one building after another? Because in the next chapter, we will meet the true masters of biological espionage, viruses that have evolved astonishing methods to silence alarms, deceive investigators, and convince an infected cell that nothing is wrong. And perhaps the most dangerous moment in any infection is not when the virus enters. It is when no one realizes that it already has. Chapter 7: How Viruses Silence the Alarm. The most dangerous spy is rarely the one carrying the largest weapon. The most dangerous spy is the one no one realizes has entered the building. By the end of our previous chapter, the infected cell had finally discovered the truth. Its molecular detectives had recognized unfamiliar genetic material. Pattern recognition receptors had identified suspicious evidence. Interferons had begun broadcasting emergency warnings. Neighboring cells were preparing their defenses. The body, at last, was beginning to fight back. If that sequence unfolded perfectly every time, viral diseases would seldom spread beyond a handful of infected cells. The story would end almost as quickly as it began. But history, whether human or biological, has always favored another kind of strategist. Not the strongest, the cleverest. Imagine a master thief entering a museum. He does not smash the display cases. He does not fire his weapon. Instead, he quietly disconnects the security cameras, cuts the telephone lines, replaces the guards' reports, changes the surveillance recordings. By the time anyone realizes the priceless artifact is gone, the thief is already miles away. Viruses have spent hundreds of millions of years perfecting precisely this strategy. They do not simply invade cells, they sabotage the investigation itself. Every alarm the cell evolved, every warning signal, every emergency response has become another obstacle that evolution has challenged viruses to overcome. This is no longer a story about brute force. It is a story about deception. The first trick is astonishingly simple. Hide the evidence. Every detective depends upon clues. Remove the clues and the detective becomes blind. Recall our previous chapter. Proteins such as RIG-I and MDA5 search for unusual forms of viral RNA, double-stranded RNA, unprotected RNA ends, molecular fingerprints that healthy human cells rarely produce. Many viruses now possess proteins specifically evolved to conceal these fingerprints. Some coat their RNA with protective proteins, disguising it from intracellular surveillance. Others keep replication hidden inside specialized membrane compartments, miniature workshops built from the host cell's own membranes. These compartments function almost like secret underground laboratories. The viral genome remains active. Replication proceeds. New viruses are assembled. Yet many of the molecular detectives never gain direct access to the evidence. Imagine counterfeit money being printed inside a hidden vault. Police officers patrol the streets. The crime scene remains invisible. Coronaviruses provide remarkable examples of this strategy. During replication, they reorganize portions of the host cell's internal membranes into enclosed structures that shield viral RNA from many innate immune sensors. The factory continues operating. The walls hide the machinery. The detectives arrive too late. Other viruses employ even more direct methods. Rather than merely hiding the evidence, they silence the alarm itself. Interferons represent one of the body's earliest emergency broadcasts. If neighboring cells receive the signal quickly enough, they prepare antiviral defenses before infection spreads. Naturally, viruses have evolved ways to interrupt that communication. Many viral proteins interfere with the complex signaling pathways responsible for producing interferons. Some prevent pattern recognition receptors from transmitting their warnings. Others disable key signaling molecules farther downstream. Still others block the activation of genes required to manufacture interferons altogether. Imagine emergency dispatch operators receiving frantic telephone calls from across the city. Unknown to them, every outgoing communication cable has already been cut. The reports never leave the building. Police headquarters hears nothing. The neighboring districts continue their daily routines, completely unaware that an invasion has already begun. Influenza viruses perform versions of this sabotage with remarkable efficiency. One of their proteins, known as NS1, interferes with several aspects of the host's antiviral response, reducing the cell's ability to generate effective interferon signaling during the earliest stages of infection. Those first few hours matter enormously. The sooner the warning is delayed, the greater the virus's advantage. Coronaviruses employ an even broader collection of proteins capable of disrupting multiple stages of interferon production and signaling. Rather than relying upon one disguise, they carry an entire toolbox. Different proteins interfere with different components of the alarm system. Redundancy increases reliability. If one trick fails, another may succeed. Then comes an even more sophisticated deception. Imagine investigators finally discovering that a building has been infiltrated. The next logical step would be to inform specialized tactical units exactly which room contains the criminals. Cells accomplish something remarkably similar. Nearly every healthy cell displays tiny protein fragments upon its surface, using structures known as major histocompatibility complex class 1, or simply MHC1. These molecular display cases constantly advertise samples of the proteins being manufactured inside the cell. Think of them as identification boards mounted outside every building in the city. Immune cells patrol continuously, reading these displays. If viral proteins appear, the building has been compromised. Specialized killer T cells receive the message. The infected cell is destroyed before the virus can spread further. It is an elegant system, unless someone removes the display boards. Several viruses have evolved precisely that solution. Herpes viruses are masters of this strategy. Certain herpes viral proteins interfere with the production, transport, or surface expression of MHC molecules. The infected cell continues producing viral proteins internally, but fewer warning signs reach the surface. The tactical units pass by. The compromised building appears ordinary. The criminals continue working inside. HIV employs comparable strategies, using accessory proteins to reduce MHC1 expression on infected immune cells. Ironically, the virus attacks the very system responsible for coordinating immune defense while simultaneously making those infected cells harder for killer T cells to recognize. It is a breathtaking example of biological espionage. Steal the uniforms. erase the records, silence the witnesses, remain invisible. But viruses face another challenge. Even if the immune system remains unaware, infected cells sometimes choose to sacrifice themselves. This process is called apoptosis. Often described as programmed cell death, apoptosis functions like a carefully controlled demolition. When severe DNA damage, overwhelming stress, or dangerous infection occurs, the cell may conclude that survival threatens the larger organism. Rather than allowing the invader to continue reproducing, it dismantles itself in an orderly fashion. Imagine discovering spies operating inside a military headquarters. The commanding officer realizes evacuation is impossible. Instead, he orders the building demolished before the enemy can capture critical intelligence. The building is lost. The nation survives. Viruses understand this threat remarkably well. Many have evolved proteins specifically designed to suppress apoptosis. Keep the cell alive. Delay its self-destruction. Extend the production schedule. Herpes viruses produce several proteins capable of interfering with apoptotic signaling pathways, allowing infected cells to survive long enough for extensive viral replication. Other viruses manipulate similar pathways using entirely different molecular tools. The objective remains identical. Do not allow the factory to close before today's production quota has been completed. Ebola virus demonstrates another extraordinary example of immune manipulation. Rather than merely infecting cells, Ebola profoundly disrupts communication throughout the immune system. Its proteins interfere with interferon responses while contributing to widespread dysregulation of immune signaling. The consequences extend far beyond the original site of infection. The emergency communication network itself begins malfunctioning. Some warnings disappear, others become chaotic. Order gives way to confusion. What emerges from all these examples is a remarkable realization. Viruses are not biologically impressive because they are physically powerful. Most are astonishingly small. Many possess only a handful of genes. Compared with human cells, they are almost absurdly simple, and yet simplicity can be deceptive. A lockpick weighs far less than a vault door. That does not make it less dangerous. Viruses succeed because evolution has refined them into specialists. Every unnecessary feature has gradually disappeared. What remains are extraordinarily efficient solutions to remarkably specific problems. How do you enter? How do you remain hidden? How do you redirect production? How do you delay detection? How do you escape before the building collapses? Millions of years of evolutionary experimentation have answered each of those questions, not through intelligence, not through planning, but through relentless natural selection. Every successful trick survived because viruses carrying it reproduced more effectively than those that did not. Generation after generation, the deception improved. The detective investigating this case eventually reaches an unsettling conclusion. The virus never intended to fight the immune system head-on. That would be foolish. The immune system is larger, more numerous, better armed. Instead, the virus changes the battlefield. It ensures that reinforcements arrive late. It conceals the evidence. It alters the reports. It persuades the defenders that everything remains under control. By the time the truth becomes undeniable, thousands of new viral particles may already have departed for neighboring cells. Perhaps this is why so many viral infections begin quietly. No dramatic symptoms. No overwhelming pain. Sometimes only subtle fatigue. A mild fever. A slight cough. The body is not ignoring the threat. It is struggling to recognize an enemy determined to erase every trace of its existence. And yet, no deception lasts forever. Eventually, enough viral proteins accumulate, enough infected cells send warnings, enough immune patrols detect irregularities. The silence begins to crack. The adaptive immune system, the body's highly specialized investigative force, is preparing to enter the story. Its arrival will transform a hidden infiltration into an open war. But before we witness that confrontation, we must remember the lesson hidden within every successful infection. Viruses are not terrifying because they are strong. They are terrifying because they are astonishingly clever. For perhaps the most frightening moment in any viral invasion is not when the virus kills a cell. It is when the cell continues living without realizing it has already been conquered. Chapter 8. When the immune system fights back. Every detective story reaches a moment when the investigation ends and the raid begins. For 7 chapters, we have followed the virus as it slipped through locked doors, forged identities, silenced alarms, and transformed healthy cells into hidden factories. It was patient, precise, almost invisible, but even the greatest criminal organization eventually makes one mistake. It leaves behind evidence: a damaged protein, an infected cell, a molecular fingerprint. One clue becomes another. One witness survives. One silent alarm finally reaches its destination. And when that happens, the body unleashes one of the most extraordinary defense systems ever assembled by evolution. It is tempting to imagine the immune system as a single army. It is nothing of the sort. It is an alliance, a vast coalition of specialists. Some patrol, some investigate, some remember, some destroy, some coordinate, others clean the battlefield after the fighting has ended. Each has a different role. Each arrives at a different time. Each fights according to different rules. Imagine an entire nation responding to a sophisticated criminal conspiracy. The local police secure the streets, detectives analyze evidence, intelligence agencies identify suspects, special forces eliminate dangerous targets, medical teams treat civilians, engineers rebuild damaged infrastructure. No single organization wins the battle alone. The immune system functions in much the same way. Among the earliest professional responders are the macrophages. Their name literally means big eaters. That description is wonderfully accurate. Macrophages wander continuously through tissues, searching for anything that does not belong: dead cells, damaged cells, fragments of bacteria, pieces of viruses. They engulf unwanted material through a process called phagocytosis, enclosing it inside membrane-bound compartments before digesting it with powerful enzymes. Imagine veteran detectives walking through a crime scene after the first explosion. They collect fingerprints, recover fragments, interview witnesses, remove dangerous debris before it harms anyone else. Macrophages perform all of those duties at the microscopic level. But they are far more than janitors. They are investigators. After digesting pieces of an invading virus, macrophages display viral fragments on their surfaces. These fragments become evidence, proof that an intruder has entered the city. That evidence will soon be presented to another extraordinary group of defenders: the T cells. If macrophages are detectives, T cells are highly trained intelligence officers and special operations forces. Yet not all T cells perform the same mission. Helper T cells coordinate the broader immune response. They analyze incoming information, issue biochemical instructions, encourage other immune cells to multiply. Without them, the immune system becomes confused and poorly organized. Killer T cells operate very differently. They search for infected cells displaying viral fragments through MHC molecules, the molecular display system we encountered in our previous chapter. When an infected cell is identified, the killer T cell approaches with astonishing precision. It does not attack neighboring healthy cells. It does not destroy entire tissues indiscriminately. Instead, it releases specialized proteins that trigger apoptosis within the infected cell. The factory is dismantled. The virus loses another production site. It is less like bombing an entire city and more like quietly removing one compromised building before the fire spreads. Meanwhile, another branch of the immune system is preparing an entirely different weapon. B cells begin transforming into plasma cells. These remarkable factories manufacture one of biology's most elegant defensive molecules: antibodies. For centuries, antibodies remained invisible mysteries. Scientists knew blood could somehow recognize familiar diseases after recovery. Only gradually did they discover these extraordinary proteins floating silently through the bloodstream. Each antibody possesses a unique shape, capable of recognizing one particular molecular target. Imagine millions of tiny detectives carrying photographs of a suspect. Most photographs are useless against any given criminal, but eventually one matches perfectly. When antibodies encounter viral particles, they attach with astonishing specificity. Some block the virus from binding receptors altogether. Others mark viral particles for destruction by macrophages. Still others activate one of the oldest biochemical defense systems in the body: the complement system. The name sounds surprisingly gentle. Its actions are anything but. Complement consists of dozens of proteins circulating quietly through blood and tissues. Most remain inactive, waiting. The moment antibodies bind to certain pathogens or alternative activation pathways detect danger, these proteins awaken one another in rapid succession, like falling dominoes, or perhaps more accurately, like a chain of emergency explosives. Each activated protein triggers several others. The cascade accelerates dramatically. Some complement proteins coat pathogens, making them easier for macrophages to recognize. Others attract additional immune cells toward the battlefield. The final components assemble into remarkable ring-shaped structures capable of punching holes through certain microbial membranes. Against many bacteria, this strategy proves devastating. Viruses often evade direct destruction because they spend much of their lives hidden inside cells, but complement remains an important ally in limiting infection and coordinating broader immune responses. By now, the battlefield has become extraordinarily crowded. Macrophages, T cells, B cells, antibodies, complement proteins. Countless other participants continue arriving. How do they avoid complete chaos? The answer lies in communication. Chemical communication. Throughout the battle, immune cells continuously release signaling molecules known as cytokines. Think of cytokines as radio transmissions exchanged among emergency responders. Some request reinforcements, others direct traffic, some stimulate inflammation, others eventually help calm the response once danger begins to pass. Every successful immune response depends upon thousands of these microscopic conversations occurring simultaneously. And suddenly, something curious happens. The infected person develops a fever. Why? For thousands of years, fever was viewed almost entirely as an illness, an unfortunate symptom, something to eliminate. Modern biology paints a far more interesting picture. Many fevers are not produced directly by viruses. They are orchestrated by the immune system itself. Certain cytokines travel through the bloodstream until they reach the brain. Specifically, regions responsible for regulating body temperature. There, they trigger a subtle adjustment. The body's temperature set point rises. Muscles begin shivering, blood vessels constrict, heat production increases. The individual feels cold, even while body temperature climbs. Why would evolution preserve such an uncomfortable response? Because many viruses reproduce most efficiently within narrow temperature ranges. Even modest increases in body temperature can slow viral replication while enhancing several immune functions. The body is deliberately changing its environment, not because the virus requested it, but because the immune system calculated that the benefits outweigh the costs. The same principle explains inflammation, swelling, redness, warmth, pain. These too often arise not because viruses physically damage tissues, but because immune cells dramatically alter blood flow and vascular permeability. Blood vessels widen, fluid enters surrounding tissues, additional immune cells flood the battlefield. Imagine firefighters breaking open walls to reach hidden flames. The building suffers additional damage, but allowing the fire to spread would prove far worse. Inflammation represents controlled destruction performed in the service of survival. This realization transforms our understanding of illness. When someone experiences sore muscles during influenza, fatigue during COVID-19, swollen lymph nodes after infection, or tenderness around inflamed tissues, many of those sensations result less from the virus itself than from the remarkable efforts of the immune system attempting to eliminate it. The heroes are fighting. Battles leave scars, yet every powerful defense carries danger. Communication systems designed for emergencies can become catastrophically overactive. Under certain circumstances, cytokines continue accumulating long after they should have been carefully regulated. More immune cells arrive. They release even more cytokines. Positive feedback develops. The process begins accelerating itself. Scientists refer to this phenomenon as a cytokine storm. The phrase is strikingly appropriate. Imagine emergency services responding to a small fire. Police request firefighters. Firefighters request military assistance. Military commanders order additional reinforcements. Roads become blocked. Communications overload. Resources intended to protect the city begin paralyzing it. The original fire becomes almost secondary. The response itself creates widespread destruction. During severe viral infections, including some cases caused by influenza viruses, SARS-CoV-2, and other highly pathogenic viruses, a dysregulated immune response can contribute substantially to tissue injury. The lungs may become overwhelmed by inflammation. Blood vessels leak. Oxygen exchange deteriorates. Organs suffer not only because viruses infected them, but because the body's own defense mechanisms have become excessively intense. This does not mean the immune system has become the enemy. Far from it. Without vigorous immune responses, many viral infections would become fatal almost immediately. Rather, evolution has optimized the system for survival across countless generations, not for perfection in every individual circumstance. Most of the time, the balance works remarkably well. Occasionally, the balance tips too far. The detective following our investigation finally recognizes the deeper truth. The body is not a battlefield divided neatly between heroes and villains. It is a living civilization attempting to preserve itself under extraordinary pressure. Every decision involves trade-offs. Destroy one infected cell to save thousands. Raise body temperature to slow viral replication. Inflame tissues to recruit reinforcements. Risk collateral damage to prevent total collapse. Victory is rarely painless. As our story moves forward, another mystery begins to emerge. Some viruses disappear completely after the immune system prevails. Others do not. They retreat, hide, remain silent for years or even decades, waiting, watching, preparing for another opportunity. Because in our next chapter, we will investigate perhaps the most astonishing escape artists in all of virology. Viruses that survive the war not by winning the battle, but by disappearing before the battle is over. And perhaps the greatest paradox of immunity is this. Sometimes the heroes save the city. Sometimes they accidentally damage it while doing so. Chapter 9. Why some viruses never leave. Every detective eventually encounters a case that refuses to close. The suspect disappears. The evidence grows cold. Witnesses stop talking. Years pass. The city returns to normal. Life moves on. Then one quiet morning, the same signature appears again, the same method, the same victim, the same invisible criminal. The investigation begins all over. For centuries, physicians faced a mystery they could not explain. A child recovered from chickenpox. The fever vanished, the blisters healed. The patient returned to school, to work, to ordinary life. Case closed. Or so it seemed. Then, decades later, sometimes 40 or 50 years later, the same person developed an entirely different illness. A painful band of blisters appeared across one side of the body. The virus had returned. But how? Where had it been hiding for half a lifetime? No detective would accept that a criminal simply vanished into thin air. Neither does biology. The truth is stranger. Some viruses never truly leave. They simply disappear from view. By now, we understand how viruses enter cells, seize molecular machinery, silence immune alarms, and produce enormous numbers of descendants. Most of those viral particles eventually encounter overwhelming resistance. Antibodies bind them. Killer T cells destroy infected cells. Macrophages clear away the debris. The visible infection ends. But evolution once again had another idea. Why fight continuously when you can simply wait? This strategy is known as latency. The word comes from the Latin latere, to lie hidden. It is one of the most sophisticated survival strategies ever evolved by any infectious agent. Imagine a master spy whose mission is no longer to steal secrets. Instead, he assumes a false identity. He purchases a small house, finds ordinary employment, lives quietly for decades. No dramatic escapes, no gunfights, no suspicious behavior. He becomes part of the city itself. The authorities eventually stop searching. The investigation fades into history. Only when the timing is perfect does the sleeper agent awaken. Certain viruses behave in almost exactly this way. Rather than producing thousands of new viral particles, they dramatically reduce or nearly stop their visible activity. Their genes remain inside the host cell. Their presence persists, but the bustling viral factory falls silent. The criminal is still inside the building. The lights have simply been turned off. Perhaps no family of viruses illustrates this strategy better than the herpes viruses. The name herpes often brings to mind only cold sores or genital infections. In reality, it is an enormous viral family that has accompanied humanity for millions of years. One member causes cold sores. Another causes chickenpox. Others contribute to infectious mononucleosis or establish lifelong silent infections that most healthy people never notice. Different diseases. One remarkable strategy. Let us begin with the familiar story of chickenpox. The virus responsible is known as varicella zoster virus, or VZV. During childhood infection, the virus spreads widely through the body. Blisters appear. The immune system responds vigorously. Eventually, the visible illness resolves. Most people assume the virus has been eliminated. It has not. Instead, a small population of viral genomes retreats into sensory neurons. These remarkable nerve cells may survive for an entire human lifetime. Unlike many other cell types, neurons divide very rarely. They are stable, long-lived, relatively protected, perfect hiding places. Inside these neurons, the viral genome remains astonishingly quiet. Only a handful of viral genes remain active. Very few proteins are produced. Almost no complete viral particles emerge. To the immune system, there is remarkably little evidence that anything unusual remains. Years later, perhaps after aging, psychological stress, another illness, or medications that weaken immunity, the balance changes. The virus awakens. It begins replicating once more. Traveling outward along nerve fibers, it eventually reaches the skin. The result is shingles, the same virus, the same person, separated by decades. The detective finally realizes that the criminal never escaped. He simply waited. Another famous member of the herpes virus family is herpes simplex virus, or HSV. Whether responsible for oral or genital infections, HSV follows a strikingly similar strategy. After the initial infection, it travels along sensory nerves until reaching clusters of neurons known as ganglia. There it establishes latency. Weeks pass. Months, years, sometimes decades. Then, under appropriate conditions, replication resumes. The virus journeys back toward the skin or mucous membranes. A familiar cold sore appears. To the patient, it feels like the infection has returned. From the virus's perspective, it never left. The pattern repeats again and again. Then comes another extraordinary relative, Epstein-Barr virus, or EBV. Most adults throughout the world carry it. Many never realize they were infected. Others develop infectious mononucleosis, the so-called kissing disease. After recovery, EBV adopts yet another version of latency. Rather than hiding primarily inside neurons, it establishes long-term residence within B lymphocytes. These immune cells normally help produce antibodies. Ironically, the virus selects members of the very system responsible for defending the body. Within these cells, EBV expresses only carefully selected genes, enough to survive, not enough to attract overwhelming immune attention. Like an undercover operative embedded inside an intelligence agency, it remains astonishingly difficult to eliminate completely. A close relative, cytomegalovirus, or CMV, pursues an equally fascinating strategy. CMV infects enormous numbers of people worldwide. Healthy individuals often experience little or no illness, yet the virus remains for life. Evidence suggests that CMV establishes persistence in multiple cell types, including cells related to the bone marrow and various immune cells. The bone marrow itself is one of the body's great manufacturing centers. Every day, it produces vast numbers of blood cells destined to circulate throughout the body. For a virus, such an environment offers remarkable opportunities. Hide among newly developing immune cells. Travel wherever those cells travel. Remain invisible until circumstances become favorable once again. Not every long-term viral survivor belongs to the herpes family. Perhaps the most famous exception is HIV. Unlike herpes viruses, HIV employs an entirely different strategy. Recall Chapter 5, where we encountered reverse transcriptase. HIV converts its RNA into DNA, but it does not stop there. Using another viral enzyme called integrase, that DNA becomes inserted directly into the host cell's chromosomes. It is no longer merely visiting. It has become part of the cell's genetic library. Some infected immune cells become activated, producing new viruses. Others enter long-lived resting states. These quiet cells form what scientists call HIV reservoirs. Inside them, viral DNA may remain transcriptionally silent for years. Modern antiretroviral therapy blocks active viral replication with remarkable effectiveness. Blood tests may detect almost no circulating virus. Yet if treatment stops, viruses emerging from these hidden reservoirs can rekindle infection. The apparent victory proves incomplete. The criminal survived inside forgotten archives. Scientists around the world continue searching for ways to eliminate these reservoirs entirely. It remains one of modern medicine's greatest challenges. These examples reveal several related concepts that often confuse even experienced readers. Latency generally refers to viral genomes remaining largely silent, producing little or no new virus while retaining the capacity to reactivate. Dormancy is often used more broadly to describe prolonged periods of inactivity, though its precise meaning varies depending on the organism being discussed. Persistence encompasses an even wider range of long-term viral survival, including situations where low-level replication continues without complete clearance. Different viruses occupy different positions along this spectrum. Some sleep deeply, others whisper. A few continue working quietly beneath the surface, but all share the same objective: survive. Not every battle must be won immediately. Sometimes patience is the greatest weapon. The detective examining these cases eventually reaches an extraordinary conclusion. The immune system is astonishingly powerful. It eliminates enormous numbers of viruses every day. It clears countless infections before symptoms even appear. Yet absolute victory is surprisingly rare. Certain viruses evolve not to overpower immunity, but to coexist with it. They reduce their visibility. Avoid unnecessary confrontation. Wait until surveillance weakens. Then resume their mission. It is less like occupying an enemy city and more like burying a sealed message beneath its foundations. Most citizens never know it exists. Time itself becomes an accomplice. This realization also explains why emotional stress, aging, serious illness, or medications that suppress immunity sometimes precede viral reactivation. The virus has not suddenly become stronger. The balance has shifted. The guards have grown fewer. The patrols less frequent. The hidden operative recognizes the opportunity, and quietly, the operation begins again. Perhaps this is the most haunting lesson in all of virology. Recovery does not always mean disappearance. Silence does not always mean absence. An invisible genome hidden within a neuron, inside a bone marrow cell, or resting within an immune cell may outlive decades of ordinary life. The story simply pauses. It does not necessarily end. Which brings us toward an even larger mystery. If viruses have accompanied living organisms for hundreds of millions of years, and if some remain with us for life, what role have they played in shaping evolution itself? Have humans merely fought viruses? Or have viruses, in subtle ways, helped shape the history of humanity? Because in our next chapter, we will leave the scale of individual cells behind and examine a far greater battlefield, one stretching across species, continents, and millions of years of evolution. For perhaps the greatest misunderstanding in medicine is this: winning one battle does not always mean the war is over. Chapter 10: The Endless Arms Race. Every mystery eventually expands beyond the crime scene. The detective who began by examining a single room suddenly discovers an international conspiracy. The clues no longer point to one suspect. They point to history itself. Until now, our investigation has remained inside a single infected cell. We watched a virus unlock the door, hijack the factory, silence the alarms, hide from the immune system, disappear into latency. But the deeper mystery has always existed beyond the microscope. Why do viruses continue changing? Why does humanity never seem to achieve a final victory? Why does every generation encounter familiar enemies wearing slightly different disguises? The answer begins with a simple observation. Neither viruses nor humans are standing still. They are running, not across cities, not across continents, across time. Imagine two master code breakers seated at opposite sides of a table. Every time one invents a stronger cipher, the other develops a better decoding method. Every improvement triggers another response. Neither side ever reaches the finish line. The contest itself becomes permanent. Biologists call this process an evolutionary arms race. Unlike the arms races of human history, however, this one did not begin thousands of years ago. It began hundreds of millions of years before the first human ever walked the earth, long before mammals, before birds, before flowering plants, before dinosaurs. Viruses already existed. Cells were already defending themselves. The first molecular battles had already begun. Every defense created pressure. Every pressure rewarded a new viral strategy. Every successful viral strategy forced hosts to evolve another defense. Generation after generation, the cycle continued. The remarkable thing is that evolution has no master plan. There is no general directing the battle, no engineer designing better weapons, only variation, selection, and time. Variation begins with mutation. We encountered mutation briefly in our previous chapter. Every time many viruses copy their genomes, tiny errors appear. A single genetic letter changes, another disappears, perhaps one is added. Most mutations accomplish nothing useful, some are disastrous. A newly produced virus may lose its ability to infect cells altogether. It quietly disappears from the evolutionary record. But every so often, a mutation changes everything. Imagine a locksmith making thousands of identical keys. One key emerges with a slightly different shape. Most altered keys fit no lock at all. One unexpected variation opens a door that none of the others could unlock. That single key suddenly becomes priceless. Evolution works in much the same way. The mutation occurs first. Its usefulness is discovered only afterward. Nature does not predict. It tests, relentlessly. This process is known as selection. Selection is often misunderstood. The environment does not create useful mutations. Instead, it quietly rewards those that already happen to work. Suppose a viral mutation allows slightly tighter binding to a cellular receptor. That virus infects cells a little more efficiently. It produces more descendants. Its genetic change gradually becomes more common, not because it was stronger, because it left behind more copies. Another mutation might help a virus escape recognition by existing antibodies. Again, descendants carrying that mutation spread more successfully. The environment performs no conscious judgment. It simply favors whatever survives. This is why variants emerge. A variant is not an entirely new virus. It is another branch on an ever-growing evolutionary tree. A collection of genetic changes inherited together. Some variants disappear almost immediately. Others spread through entire populations. Most are biologically unremarkable. A few alter the course of history. Humanity witnessed this process unfold in extraordinary detail during the COVID-19 pandemic. As SARS-CoV-2 circulated through millions of infected individuals, countless mutations accumulated. Most vanished. Some persisted. Occasionally, combinations of mutations improved transmission or altered immune recognition sufficiently to become dominant variants. For perhaps the first time in history, billions of people watched viral evolution happening almost in real time. Yet SARS-CoV-2 is hardly unique. Influenza viruses have performed similar evolutionary dances for centuries. Their remarkable ability to change surface proteins helps explain why influenza vaccines often require regular updates. The detective studying these cases eventually notices an uncomfortable pattern. The criminal keeps changing its appearance. Every time investigators memorize one face, another appears. But evolution tells only half the story. The other half belongs to the hosts. To us, humans evolve far more slowly than viruses. A virus may complete thousands of generations while one human generation passes. From that perspective, the contest appears hopeless. How could an organism reproducing every few hours lose against one reproducing every few decades? The answer lies in cooperation. An individual human changes very little genetically during life. Human populations, however, possess another remarkable advantage. The immune system learns. Every infection leaves behind memory B cells, memory T cells, antibodies refined through previous encounters. One person may recover. The next exposure triggers a faster response. Communities gradually accumulate immunity. Vaccines accelerate this process dramatically by teaching the immune system without requiring severe disease. Unlike viral mutations, immune memory does not alter DNA passed to future generations. Instead, it transforms the defenses of each individual. Evolution and learning work together. Different timescales, same battle. Meanwhile, over much longer periods, natural selection has shaped humanity itself. Certain genetic variations provide increased resistance to particular viral infections. The CCR5.32 mutation we encountered earlier reduces susceptibility to many HIV strains. Other genetic differences influence interferon signaling, immune recognition, or inflammatory responses. Viruses shape hosts. Hosts shape viruses. The contest continues. Then comes one of evolution's greatest mysteries. How do entirely new viral diseases suddenly appear in humans? The answer often begins with what scientists call a cross-species jump, or more broadly, zoonosis. Imagine a key carefully crafted for one particular lock. Year after year, it fits perfectly. One day, a tiny manufacturing error changes its shape. Unexpectedly, it also fits another lock. Not perfectly, but well enough. Many viruses circulate harmlessly or with limited effects inside their natural animal hosts. Bats, birds, rodents, primates, livestock. Generation after generation, they remain confined within those species. Occasionally, mutations alter viral proteins just enough to recognize receptors in another species. Perhaps a hunter handles infected wildlife. Perhaps domestic animals serve as intermediaries. Perhaps ecological changes bring species into closer contact than ever before. The opportunity appears. The virus enters a new host. Most attempts fail immediately. The species barrier remains remarkably effective. But every so often, the fit proves sufficient. Replication begins. Transmission follows. A new chapter of biological history opens. Many of humanity's most significant viral diseases trace their origins to such events. Influenza viruses circulate among birds and other animals before certain strains adapt to humans. HIV emerged following cross-species transmission of simian immunodeficiency viruses from non-human primates. Coronaviruses capable of infecting humans have evolutionary histories involving animal reservoirs. Different viruses, different pathways, one recurring principle. Nature constantly conducts experiments. Most remain invisible. Occasionally, one reaches us. If local transmission continues successfully across many individuals, a regional outbreak may emerge. If international travel, dense populations, and efficient human-to-human spread combine, the result can become a pandemic. The word itself comes from Greek roots meaning all people. History remembers these moments vividly, not because viruses suddenly became evil, but because ancient evolutionary processes intersected with an increasingly connected human civilization. A mutation occurring inside one host can eventually influence every continent. The detective investigating this immense conspiracy eventually realizes that no single villain exists. Evolution itself is the author. Viruses do not plan pandemics. They do not seek world domination. They possess no ambitions, only genomes that continue changing. Natural selection performs the editing. Human behavior influences the opportunities. Ecology shapes the encounters. History records the consequences. And through it all, another race continues quietly in the background. Every new viral strategy pressures immune systems. Every improved immune defense pressures viruses. Some mammals evolve stronger antiviral proteins. Viruses evolve methods to disable them. Hosts alter receptors. Viruses adapt binding proteins. Hosts improve immune recognition. Viruses conceal their molecular fingerprints. The cycle repeats. Millions of years pass. No final victory arrives. Perhaps this explains one of biology's deepest truths. Life is not defined by stability. It is defined by adaptation. The detective searching for the end of the story discovers instead that there is no ending. Only another chapter, another mutation, another adaptation, another generation learning from the last. Humans are remarkable because we possess something evolution alone never created: science. Instead of waiting thousands of generations for genetic changes, we develop vaccines, antiviral medicines, genomic surveillance, molecular diagnostics, artificial intelligence capable of detecting evolutionary patterns. Knowledge becomes another form of adaptation, perhaps our most powerful one. Yet even science cannot abolish evolution. It can only learn to move with it. The race continues. The players change, the rules evolve, the contest endures. Which brings us toward the final questions of our journey. Can humanity ever truly outsmart viruses? Will future medicine finally break this ancient cycle? Or are viruses destined to remain permanent companions of life itself? Because in our next chapter, we leave the distant past behind and turn toward the future, a future shaped by mRNA vaccines, antiviral drugs, CRISPR, synthetic biology, and technologies that previous generations could scarcely imagine. For perhaps the greatest lesson hidden within this endless race is not that viruses always change. It is that life, in every form, never stops adapting. And perhaps the most humbling realization of all is this: we are not the first opponents viruses have ever faced, and we will almost certainly not be the last. Chapter 11. Can we outsmart viruses? Every great detective story ends with the same question. Now that we understand the criminal, can we stop the next crime before it happens? For thousands of years, humanity fought viruses almost blindly. We knew the symptoms, we feared the epidemics. We watched entire civilizations disrupted by invisible enemies that no one could even see: smallpox, polio, influenza, measles, rabies, more recently, HIV, Ebola, COVID-19. Each generation inherited a familiar feeling. We were always reacting. The virus moved first. Humanity responded afterward. But something extraordinary has happened over the past century. For the first time in history, we are beginning to understand the virus not merely as an enemy, but as a sequence of predictable molecular events. And every predictable step offers an opportunity for intervention. Think back to the beginning of our investigation. The virus did not conquer the cell in one dramatic leap. It followed a sequence. First, it found the correct receptor. Then, it entered the cell. It removed its protective coat. It copied its genome. It hijacked ribosomes. It assembled new viral particles. Finally, it escaped to infect another cell. Imagine watching a master thief rob a bank. He studies the entrance, disables the alarm, opens the vault, collects the money, escapes through a hidden tunnel. If investigators understand every stage of the robbery, they no longer need to stop the thief at only one moment. They can interrupt the plan almost anywhere. Modern antiviral medicine follows exactly this philosophy. Rather than attacking viruses indiscriminately, scientists ask a far more precise question. Which step can we block? The earliest opportunity comes before the virus has even entered the cell. Entry inhibitors prevent viruses from attaching to or penetrating their target cells. Without access to the correct receptor, the invasion never begins. A remarkable example comes from HIV therapy. Certain drugs block the CCR5 receptor used by many HIV strains. Imagine replacing every lock in a city overnight. The master key carried by the intruder suddenly fits nothing. Other experimental approaches attempt to interfere directly with viral attachment proteins instead. Different strategy. Same objective. Keep the door closed. Suppose the virus succeeds in entering anyway. Another opportunity appears. Uncoding inhibitors prevent the viral capsid from opening correctly. Recall Chapter 3. After entry, the viral genome remains imprisoned inside its protective protein shell. Only after uncoding can replication begin. If the capsid refuses to open, the instructions remain locked away. The factory never receives its counterfeit blueprints. It is rather like capturing a spy who carries an encrypted briefcase that no one, including the spy, can unlock. The mission ends before it begins. But perhaps the virus has already escaped its capsid. Now its genome must be copied. This stage has become one of the most successful targets in antiviral medicine. Different viruses require different enzymes. RNA viruses often depend upon RNA polymerases. Retroviruses rely upon reverse transcriptase. DNA viruses use their own specialized replication machinery or exploit host enzymes. Drugs that inhibit these enzymes are known broadly as polymerase inhibitors or reverse transcriptase inhibitors, depending upon the specific target. Imagine sabotaging the printing presses inside a counterfeit currency operation. The workers remain present. The building remains intact, but no additional copies can be produced. Replication slows dramatically. Some infections become manageable. Others become nearly undetectable. Another remarkable target appears slightly later. Many viruses manufacture enormous chains of proteins that must be cut into smaller functional pieces. This task is performed by enzymes known as proteases. Without them, viral proteins remain unfinished. Useful components never emerge. Scientists developed protease inhibitors to exploit this vulnerability, particularly in HIV treatment. Imagine an automobile factory producing only complete sheets of metal because every cutting machine has failed. The raw materials exist. Finished cars never appear. The viral assembly line collapses. Replication has occurred. Proteins have formed. Capsids assemble. Yet one final obstacle remains: escape. Newly manufactured viruses must leave the infected cell before they can spread elsewhere. Some antiviral drugs interfere with this process. One famous example involves influenza. Influenza viruses use an enzyme called neuraminidase to help newly formed viral particles separate from infected cells. Neuraminidase inhibitors reduce this release. The viruses accumulate. Their expansion slows. Imagine police surrounding every exit of a warehouse. The criminals remain inside. Their operation cannot expand into neighboring buildings. The strategy is containment. When viewed together, these therapies reveal something fascinating. Modern antiviral medicine rarely attempts to kill viruses directly. Strictly speaking, viruses are not fully alive in the traditional cellular sense. Instead, medicine interrupts the sequence of theft: entry, uncoding, replication, protein processing, assembly, release. Break enough links in the chain, and the entire operation fails. Yet despite these remarkable advances, antiviral therapy remains profoundly more difficult than treating bacterial infections. Why? The answer returns us to one of the central mysteries introduced in Chapter 1. Bacteria are complete living cells. They possess ribosomes unlike ours, distinct cell walls, unique metabolic pathways, different enzymes. Antibiotics exploit these differences. They target structures human cells either lack or use differently. Viruses offer far fewer opportunities. They carry astonishingly few genes. Most of the machinery they use belongs not to the virus, but to us. The ribosome producing viral proteins is the cell's own ribosome. The ATP supplying energy belongs to the host. The membranes surrounding viral replication often originate from host cells. Attacked too aggressively, and we risk damaging healthy tissues alongside infected ones. Imagine trying to stop counterfeit documents being printed inside your own government offices. Destroy the printing presses, and legitimate work stops as well. Precision becomes absolutely essential. Another challenge follows naturally. Viruses evolve rapidly. Every replication cycle introduces opportunities for mutation. A single altered amino acid may reduce drug binding. The medicine becomes less effective. Drug resistance emerges. This is why many viral diseases require combinations of antiviral drugs rather than relying upon a single therapy. HIV treatment transformed medicine precisely because multiple drugs attack different stages of the viral life cycle simultaneously. Escape becomes dramatically more difficult. The thief now faces locked doors, disabled vehicles, sealed tunnels, and surveillance cameras all at once. Modern science, however, is reaching far beyond traditional antiviral drugs. Perhaps the most revolutionary development of recent years has been the rise of mRNA vaccines. For generations, vaccines often relied upon weakened viruses, inactivated viruses, or purified viral proteins. mRNA technology approaches the problem differently. Rather than delivering the viral protein itself, it delivers temporary genetic instructions telling human cells how to manufacture one carefully selected viral protein. The immune system studies that harmless protein, builds antibodies, develops memory T cells, then the instructions disappear. When the real virus eventually arrives, the immune system recognizes it almost immediately. The detective has already seen the suspect's photograph. Another exciting frontier involves monoclonal antibodies. Instead of waiting for each patient's immune system to produce effective antibodies, scientists manufacture highly specific antibodies in the laboratory. These engineered molecules can bind viral proteins directly, blocking infection or helping immune cells eliminate viral particles more efficiently. Although some viruses evolve around them over time, monoclonal antibodies have demonstrated remarkable value against several infectious diseases. Yet perhaps the boldest ideas belong to the future. Imagine editing the criminal's instruction manual itself. This is the promise, still developing, of CRISPR and related gene editing technologies. Originally discovered as part of bacterial immune defenses against viruses, CRISPR functions like extraordinarily precise molecular scissors. Scientists are investigating ways to use these systems against viral genomes, particularly those hiding inside infected cells. The challenges remain enormous: delivering CRISPR safely, avoiding unintended genetic changes, reaching every infected cell. But the concept itself is breathtaking. Rather than merely slowing viral replication, erase the hidden instructions altogether. Researchers are also pursuing broad-spectrum antivirals, medicines capable of working against many unrelated viruses instead of only one. Others investigate host-targeted therapies, subtly modifying cellular pathways that numerous viruses depend upon while preserving normal cell function as much as possible. These strategies reflect an important realization. Perhaps the most reliable way to defeat many viruses is not by chasing each individual criminal, but by strengthening or protecting the buildings they all attempt to exploit. The detective following our journey now understands something that seemed impossible back in the prologue. Viruses are not magical. Every successful infection follows rules. Every rule creates a vulnerability. Every vulnerability offers another opportunity for science. Some opportunities become vaccines, others become antiviral drugs. Still others remain waiting inside laboratories across the world, hidden within experiments whose full importance we do not yet understand. Will humanity ever eliminate every virus? Almost certainly not, nor should that necessarily be our goal. Many viruses coexist harmlessly with humans. Some may even influence ecosystems in ways we are only beginning to appreciate. The true objective is far more practical. Understand, predict, interrupt, prevent unnecessary suffering. Our greatest advantage has never been physical strength. It has always been knowledge. Every chapter of this investigation has revealed another layer of an astonishing molecular conspiracy. We now know how viruses locate their victims, how they unlock cells, hijack ribosomes, silence alarms, evade immunity, hide for decades, evolve across generations, and now, how science is gradually learning to interrupt each step of that extraordinary process. But one final mystery remains. If viruses are neither fully alive nor completely inert, if they have shaped evolution for hundreds of millions of years, what do they ultimately reveal about life itself? Because in our final chapter, we will step beyond medicine, like beyond virology, and beyond the infected cell to confront the deepest question of all. Perhaps viruses are not merely biological thieves. Perhaps they are among the greatest teachers life has ever known. For the only way to truly outsmart a virus is to understand every single step by which it quietly steals a living cell. Chapter 12. The Cell That Saved Humanity. Every mystery must eventually answer one final question. Not who committed the crime, not how it happened, but why the story matters. Our investigation began with an ordinary cell. It was not heroic. It did not know history. It did not know that an invisible particle was drifting toward its surface. It simply carried out the quiet work that every healthy cell performs every second of every day. Producing proteins, generating energy, repairing damaged DNA, listening to chemical messages from neighboring cells, maintaining the extraordinary civilization we call the human body. Then the virus arrived. There was no explosion, no warning siren, no dramatic invasion. Only a microscopic particle so small that millions could fit upon the head of a pin. Within minutes, the cell's own machinery had been turned against it. The factory no longer served itself. It served the invader. From that single moment, an extraordinary detective story unfolded. We followed the virus as it searched for the perfect receptor. We watched it unlock the cell without breaking down the door. We uncovered the astonishing deception that allowed ribosomes to manufacture viral proteins without realizing they had changed employers. We entered hidden replication factories, where one viral genome became thousands. We witnessed the first desperate alarm signals carried by interferons. We watched molecular detectives search for evidence that something had gone terribly wrong. We discovered how viruses concealed their fingerprints, silenced warnings, and hid inside neurons, immune cells, and bone marrow for decades. We followed humanity's response through vaccines, antiviral medicines, monoclonal antibodies, and gene editing technologies that previous generations could scarcely have imagined. Each chapter seemed to reveal another ingenious strategy, another hidden passage, another forgotten clue. And yet, perhaps the greatest discovery was not about the virus at all. It was about the cell. At first glance, a single cell appears astonishingly fragile. It cannot outrun a virus. It cannot reason. It cannot consciously defend itself. Left entirely alone, an infected cell often loses. Sometimes within hours, sometimes even sooner. If the story ended there, life on Earth might never have survived. But the story never belonged to one cell. It belonged to billions, then trillions. Your body is not a single organism in the way we often imagine. It is a living society. Roughly 37 trillion cells cooperate continuously. No parliament directs them. No president commands them. No central engineer oversees every decision. Yet somehow they communicate, coordinate, repair, defend, and adapt with astonishing precision. Imagine the largest city ever built. Every citizen performs a specialized occupation. Some generate energy, some transport oxygen, some construct connective tissues, some conduct electrical signals. Others patrol continuously searching for danger. No individual citizen understands the entire civilization. Yet together, the civilization survives. The immune system represents perhaps the greatest example of this cooperation. A lone macrophage cannot eliminate every virus. A single natural killer cell cannot patrol the entire body. One antibody accomplishes little by itself. One T cell cannot monitor 37 trillion neighbors. Their strength emerges only through collaboration. Signals pass from cell to cell. Interferons spread warnings. Cytokines coordinate responses. Memory cells preserve experience. Bone marrow replaces exhausted defenders. Lymph nodes become meeting places where evidence is examined and strategies refined. The body succeeds not because any individual component is perfect, but because countless imperfect components work together. Perhaps this is evolution's greatest lesson. Complexity creates resilience. Throughout this podcast, we often described viruses as master thieves, spies, hackers, and code breakers. Those metaphors reveal something profound. Viruses force life to improve. Without viral pressure, many of the immune defenses we now take for granted might never have evolved. Pattern recognition receptors, interferons, natural killer cells, adaptive immunity, antibodies capable of extraordinary precision. Even parts of our own genomes carry ancient viral fingerprints. Modern genetics has revealed that fragments of viral DNA became permanently incorporated into the genomes of our ancestors millions of years ago. Most remain inactive. Some have acquired entirely new purposes. One remarkable example involves proteins essential for the formation of the human placenta. Evidence strongly suggests that these proteins originated from ancient viral genes that evolution repurposed for an entirely different role. The former invader became a collaborator. The thief became a builder. History, it seems, rarely divides neatly into heroes and villains. Evolution is more creative than that. Viruses have caused immeasurable suffering throughout human history. Pandemics have reshaped civilizations. Families have been devastated. Entire generations have lived beneath the shadow of invisible epidemics. Those realities must never be minimized. Yet at the same time, viruses have also acted as relentless teachers. Every infection challenged life to develop stronger defenses. Every epidemic accelerated scientific understanding. Every mystery forced another generation of researchers to ask better questions. The history of virology is not merely the history of disease. It is the history of discovery. Think of the extraordinary technologies humanity has created in response. Vaccination transformed medicine long before anyone had seen a virus under a microscope. Molecular biology revealed how genetic information flows through living cells. Polymerase chain reaction made invisible genomes detectable within hours. Genome sequencing now allows scientists to observe viral evolution almost in real time. mRNA technology demonstrated that temporary genetic instructions could safely train the immune system with breathtaking speed. Artificial intelligence increasingly assists researchers in predicting protein structures, identifying drug candidates, and monitoring emerging variants across the globe. The detective story continues. Only now, humanity has become a far better investigator, and still, science remains humble. Every answer uncovers another question. How do some viruses establish perfect latency? Can HIV reservoirs one day be eliminated completely? Will broad-spectrum antivirals protect against future pandemics? Can CRISPR safely remove persistent viral genomes from infected tissues? How should we prepare for viruses that have not yet crossed into humans? The future of medicine will almost certainly depend upon answering questions we have not yet learned to ask. But perhaps our greatest advantage has never been technology alone. It has always been memory. Not merely human memory preserved in books, laboratories, and universities. Biological memory. Immune memory. The remarkable ability of the adaptive immune system to remember a microscopic encounter long after the danger has passed. When a virus infects you for the first time, your immune system is learning an unfamiliar language. It experiments, it makes mistakes, it improves. Specialized B cells refine antibodies with extraordinary precision. Memory T cells preserve successful strategies. The next encounter unfolds differently. Recognition comes faster. Communication becomes more efficient. The response grows stronger. In many cases, the virus never again gains the same advantage. Vaccines harness this remarkable principle without requiring the body to endure the full consequences of severe disease. They are not magic. They are education, a carefully prepared lesson delivered before the examination begins. Perhaps that is the most beautiful idea in all of immunology. The immune system is not simply a weapon, it is a student. Every infection becomes a lesson. Every successful recovery becomes accumulated knowledge. Every generation of scientists adds another chapter to humanity's own memory. And so we return to the ordinary cell with which our story began. The cell that lost its factory, the cell that sent an SOS, the cell that sacrificed itself so that neighboring cells could prepare. Alone, it could never defeat the virus. It was never expected to. Its purpose was not solitary victory. Its purpose was contribution. One warning, one signal, one act of resistance passed forward through an unimaginably vast biological community. That is how living systems endure. Not because every individual survives, but because information survives, knowledge survives, memory survives. Long after one infected cell has disappeared, its warning echoes through the immune system. Long after one epidemic has ended, humanity remembers. Long after one generation of scientists retires, another continues the investigation. Perhaps that is why viruses have never achieved their ultimate victory. Not because they lack intelligence, not because they are weak, but because life itself possesses an extraordinary ability to learn. Every recovered patient carries invisible evidence of that lesson. Every vaccine extends it. Every scientific discovery strengthens it. Every child born into a world protected by generations of accumulated knowledge becomes part of a story that began hundreds of millions of years ago. And that story is still being written, at this very moment, while you are listening to these words. Trillions of cells inside your body are communicating. Proteins are folding. DNA is being repaired. Immune cells are patrolling. Memory cells are waiting. Somewhere, perhaps, a single virus has already entered a cell. Somewhere else, another infected cell has already raised the alarm. Some battles are beginning, others are ending. Most will unfold so quietly that you will never know they happened. And perhaps that is the greatest triumph of all, because every time you recover from a viral infection, your body does more than heal. Your immune system has learned something new. It has added another page to an invisible encyclopedia written not with ink, but with living cells. An encyclopedia that has protected our species through countless epidemics, changing climates, mass extinctions, and millions of years of evolution. The silent war continues every second. Inside every heartbeat, inside every breath, inside every living cell. Not as a story of fear, but as a story of resilience. A story that began long before humanity existed, and one that will continue long after this podcast comes to an end. If you enjoyed joining this slow journey into the hidden world inside every cell, we would truly appreciate your support. Our channel is still in its early stages, and every like, every subscribe, and every share genuinely helps us continue creating thoughtful, science-based podcasts like this one. If there was a moment, a question, or a scientific mystery that stayed with you, we'd love to hear about it in the comments. And if there's another topic in biology, medicine, evolution, space, history, or any other scientific mystery you'd like us to explore in a future podcast, please let us know. Your ideas often inspire the next investigation. Thank you for spending your time with us. Until our next journey, stay curious. Because somewhere inside you, the greatest detective story in nature is still unfolding.